So welcome everybody in this nice cold room. I am Hélène SOUBELET. I am the director of FRB, which is a foundation for research on biodiversity, French foundation. So on behalf of Professor Découvrez, which is the president of FRB, I would like to warmly thank you to be there. To thank the CNRS for hosting us this afternoon, to thank the speakers, of course, and our other financial partners, which are CMEB, Muse, and EDF Group. So, as you may know, FRB is a French foundation for scientific cooperation. It is funded by the main French research institute on biodiversity and the Ministry of Research. And our mission is first to support different forms of research, such as the CESAB, synthesis project, but also other innovative forms of research, such as the systematic review of scientific literature. And second, our mission is to promote the dialogue between scientists and stakeholders, public servants, for example, private company, but also NGOs. And third, our last but not least mission is to communicate widely on the results of research and to promote those results. So I am also happy today because this is the free program research of CESAB is the first program since that have a very strong link with a private partner, another partners, so EDF Group. This research program was selected in 2015. And it started only in 2018. And thanks to the financial support and the scientific involvement of EDF, represented here by Anthony Maire and Jérémy Butin, this cooperation has proven that the concept works. So we can lead research with private or public companies. And FRB is very delighted for that because this is our main purpose. I am also happy because this is the first CESAB conference since November 2019. And in this last conference, we learned about challenge and opportunities for large-scale biodiversity conservation with the pelagic group. So this is the free group, another CESAB group, and this time we will focus on from species to functions toward a paradigm shift for biodiversity conservation, and we will focus on biological rarity, so the functional rarity. And I am looking forward myself to learn more about it because I know that the free group has worked on the conceptual basis of the concept and has proposed a global analysis in order to identify areas of major importance due to rare species. I think that what is rare is irreplaceable once lost. So I think this places humanity in front of its responsibilities to conserve the species, their habitats and the ecosystems. And I think to do that, research is very crucial. And I hope we will see that this afternoon. And FRB is proud to be part of it. So now I will leave the floor to Nicolas MOUQUET, which is the scientific director of CESAB. Thank you very much. Have a good conference.
Yeah, hi everyone. So I'm Nicolas MOUQUET, the scientific director of the CESAB. I'm also a researcher with the CNRS. And I'm very glad to be here and to see humans again. It's good to have you all in person. So the CESAB have moved to Montpellier a few years ago, 3 years ago actually, and we are downtown Montpellier at the Institut Buisson-Bertrand in front of the old University of Medicine. So the CESAB is a tool developed by the FRB 10 years ago to give researchers some time for large-scale biodiversity analysis. So what do we mean by synthesis? Well, to make it simple, to quote Edward O. Wilson, synthesis is simply the art of putting together the right information at the right time and think critically about it. We, in biodiversity science, we see two kinds of synthesis, so consensual synthesis and exploratory synthesis. So to make it more clear, on that figure, if you plot on the x-axis the level of agreement within a community of researchers, and on the x-axis the quantity and the quality of evidence, well, when those two measures are strong or high, then we can assume that there is consensual synthesis, and this is what is made in, for instance, IPBES reports that you know and that are targeted toward decision. But for the rest of that uncertainty space, this is what we call exploratory synthesis. And this is where we need a synthesis center to help increase the level of agreement within the community and increase the quantity and the quality of the evidence to switch from the exploration to the consensus. So to do so, the CESAB offer researcher a place and some time to assemble and analyze existing data on biodiversity, to conduct analysis and address the major scientific and societal challenges of biodiversity conservation, and to contribute to new conceptual and methodological advances in biodiversity science. Initially, the CESAB was simply based on funding a group of up to 10 or 15 researchers for 3 years. And those people would come to the CESAB twice a year. They would be helped with a postdoc for 24 months. And they would all together work on the synthesis on any kind of topics. And when we moved to Montpellier, we've been challenging and evolving that model. And we split our focus to fund small groups or large groups and also thematic groups, and open, what we call open groups, like groups that would be funded on whatever topic they would like to study. And by radiating that funding opportunities, we managed to increase the number of partners we could work with and increase the number of groups that have been funded by the CESAB. This increase illustrates the momentum that we are living right now on synthesis and biodiversity. There is a need for that. We know it by heart. But also it illustrates the quality and the capacity of the French community to be leader on that particular topic. And there are a very much reason to be proud of that increase. Those are the groups actually funded at the CESAB, so I won't go into the details. They are studying very different topics, from fundamental questions such as the evolution of the reproductive strategies in plants to very applied topics such as the power of decision within groups working on conservation biology, also justice of conservation. So it's a mix between fundamental science and applied science. We are funding at the CESAB right now. And this is the active groups, but we actually do follow the groups when they have ended their research project. And this is what happened with the free working group, which you don't see there because it has ended this year. And that group is what I call a meta group. So it is led by Cyrille VIOLLE, and as as Hélène said, it's working on functional rarity. It's an ecosystem. It's not a group anymore. It's an ecosystem of researchers that took advantage of the CESAB to build a network of collaborators to develop some satellite project funded by some other means. And they've been using the CESAB as a basis to grow and become bigger and bigger. And this year they could come back on their own funding, thanks to EDF and the FRB, for a week of workshop here. But they've been organizing that fantastic conference that we will have this afternoon. So I want to thank them very much. They also organized a workshop this morning for students on functional rarity packages in R. So it's really a matter for that group to give back to the community what they received from their funders. So I'd like to thank them very much. So now I'm gonna self-promote a little bit. In 22, the CESAB will be still very active on the side of funding. So this year, we have opened 3 calls for different projects, and 8 new groups will be funded, which is a lot, given the CESAB standard. And 2 of those calls just opened a few weeks ago. So I'd like you to take the opportunity to consider applying to those calls. So the first action is a new action we are providing at CESAB. It's called Data Share. It's a joint call that we could organize thanks to the partnership with Biodiversité and the LabEx Tulip. And the main objective is to accelerate the sharing of open access and large-scale biodiversity-related data set. And that proposal, that call is new for us. So we've been building up all from the scratch. And I hope that we will get a lot of proposals this year, which will make it worth doing it again next year and the year after and after. And it's really a paradigm shift also in synthesis to disconnect, the development of database from the analysis of the data. I'm not saying that we have to build and provide data without any purpose, but we understand now that those are that might be 2 different objectives, and we have to make a lot of energy to help researchers sharing the data and making the sharing reproducible. And the other call we opened is it's a collaboration again with the Minister of de la Transition écologique and the French Foundation of Biodiversity. It's not foundation, sorry. But whatever. That call is on the impact of the terrestrial biodiversity in the Anthropocene. It aims to measure, identify, and monitor the influence of human activities on biodiversity and the best practices to be promoted. The pre-proposal deadlines are on the 22 of September. And to make it short, we already opened that call last year, and we haven't received that much proposals. So I really suggest that you go on the website and check if you can apply. Don't hesitate to give a try. And finally, that fantastic science ecosystem is used to fuel training programs that we think are very important destination to the young scientists. And this month's registration are open for 2 weeks of training courses that will be held this fall. So one is organized on biodiversity knowledge synthesis, an introduction to meta-analysis and systematic reviews. And the other is on reproducibility in research for computational ecologists. And they combine top scientific teaching, and we made them more than affordable. Our aim here is only for the science going on at the CESAB to fuel within the next generation of ecologists. Okay, that was the self-promotion section. And now it's time for me to thank the people without which this conference would not have been possible. So obviously Cyrille VIOLLE, PI of the free group, but also Anthony Maire, so he's a researcher from EDF who joined the group at the beginning, and he really made me believe in that partnership. He's been giving us a lot of insight on the scientific side, and we've been working together, and that was a great experience. Thank you very much, Anthony. Obviously, the staff from FRB, Marie-Claire Danner, Maud Calmet, and Nicolas Casajus, who made all that possible. I'd like to thank also the people who have organized the workshop this morning, so Nicolas Casajus, but also Sébastien Légier, and Mathias Cornier, and Camille. I'm very sorry, Camille, I forgot your last name, but Camille, thank you so much. You're here, Camille. And I'd like also to thank the CESA postdocs who will be chairman this afternoon. Thank you all for being here. And now it's I give the stick to Cyrille VIOLLE, who will introduce the conference. Thank you.
So yeah, thank you very much to be there. It's really nice to have a conference in person, finally. So yes, I'm Cyrille VIOLLE. I'm the PI of the free working group. So who knows the CESAB in this room? You can more or less add. If you don't know the CESAB and synthesis centers, you should apply. For me, no, I'm serious. For me, it's really the most efficient and stimulating research tool we have. For us, we spend 1 week, twice a year, together in a building, outside labs, almost outside image, not always, but it's really efficient. Something happens. You are doing new things. So at least we try. So yes, so we have a working group that is called FREE, so for functional rarity in ecology and evolution. And so we start this working group indeed more or less 4 years ago, and it's really a research frontier where we try to develop a new concept. Also, at the meantime, we try to explore new data to test this concept. I think we discovered some new patterns at local and also global scales. And in the meantime, we try to develop theories around this concept. And so we really had a chance that will be support on the long term. So we will go on this adventure by really looking now at some implications of our findings and our concept for conservation, but also for ecosystem functionings and nature's contribution to people. And another objective now is to see whether and how we can have some applied perspectives of rarity and functional rarity. And of course, we'll try to make predictions and theoretical predictions. So for that, we have quite a big group, maybe a bit above 15, in which we try to put together experts in theoretical ecology, functional ecology, community ecology, conservation ecology, microbiology, evolutionary biology, but also biogeography. And so we also had the chance to have experts from different taxonomic groups. So the idea is to see whether and how some patterns we found are common to different taxonomic groups: plants, microbes, fishes, mammals, birds, insects, reptiles, and others. We also benefit from the rise of lots of thread database for the last decades. Only a few examples, but now you have tens, maybe hundreds of database. So we have been able to compare different taxonomic groups. Here is just an example of thread covariations, what we call functional thread space, across taxonomic groups, plants, mammals, microbes, and so on. And we start to find common patterns that could be interesting to tackle from both macroecological and evolutionary questions. what we are asking in the group is to try to understand the causes and the consequences of rare phenotypes in populations, communities, and beyond from very different perspectives from theory, for instance, by applying a theoretical corpus of evolutionary biology, to global analysis of diversity, trait diversity, and functional rarity, up to really applied perspective in crop diversifications, for instance, to test whether and how we can mix different species or different genotypes of a given species, and if this is really important to account for some rare phenotypes in the field. But of course, because we are talking about rarity, our big question is whether it matters for conservation. So this is why I'm really proud today of this conference, in which you will have different talks from internal and external people to the group interested by conservation and functional ecology and macroecology: Ana Rodriguez, Brian Enquist, Catalina Pimiento, and David Mouillet. Just in terms of organizations, we'll have a break between Brian and Catalina's talk, so it will be in the hall and outside. And also at the end of the conference, around 5:36 p.m., something like that, we offer you a cocktail for all of you in the hall of the Cefe. So it's the lab close to this amphitheater. But you will see. You can follow the group. Of course, I would like to thank a lot of people for this conference and for the group. So first, the LabEx CEMEB and Muse to have funded part of these conferences, especially the cocktail. The CNRS, because we can use this amphitheater thanks to them. And the Cefe to use the hall for the cocktail. Of course, Marie-Claire and Maud, I don't know where they are. Thank you so much for this organization, for the conference, for everything, for the group, for organizing our life. You are great. And obviously, this group is a really special partnership between FRB and private companies, so EDF. So it's really special for me. So when I start to talk to EDF, I say, well, how I will explain what we are doing, does it interest them? And in fact, we talk only about research, never money. We talk about research. And even this partnership is so special because in our group, as a core member of our group, we have one researcher from EDF, Anthony Maire, with whom we are doing science all the time. And we have the same questions, same interests. So it's really, I think it's really not unique, interesting example anyway of partnership between academic science and a private company. So thank you very much. And now I would like to introduce Jérémy Butin, who is the representative of EDF.
Well, good afternoon, everybody. I'm Jérémy Butin, and I'm the manager of the EDF R&D team dedicated, among others, to ecology, to research in the field of ecology. So first of all, on behalf of all EDF and its R&D division, I thank you for the invitation. I thank the organizer for having us here in the warm city of Montpellier, as you can see. So unfortunately, I'm not going to talk about functional rarity today or about money. I promise, but just to give some. So I think it's okay. But I wanted to seize this opportunity to explain a little bit what are our, as a company or responsible company, I think, what are our environmental challenges and how we are organized within EDF R&D to deal with them. So for us, environment is two very different things. It can be something that can interact or interfere with our capability of produce safely electricity, Such interference could come from a lot of different things. Some are very obvious, such as meteorological extreme events, floods, storms, or on the contrary, low water resources. We need water to cool down our nuclear power plants, as an example. It could come from clogging of water intakes due to ice, sediment, living organisms sometimes. And in the meantime, we have regulation evolution all the time, safety constraints, which are getting harder to reach, which is a good thing. And we have climate change that is modifying the occurrence or the frequency of such external environmental risk and extreme events. So we have to be organized, we have to model, to anticipate, we have to adapt processes or design for new facilities. We have to change operating parameters just to adapt to both regulation evolution and external environmental risk within a global changing environment. But environment is obviously not only an aggression, but it's something towards we have a responsibility and we have to limit and mitigate our impacts. So such impacts coming from EDF, I mean, can be coming from our carbon emissions, because even though we have low carbon emissions power plants, mainly, we still produce some CO2. We are using water, as I explained before, and we are releasing warmer water in the environment after having cooling down our nuclear power plant, as an example. We are forced to have a little bit of chemical or radiological releases. When we build something, it leads to soil artificialization, modification of habitat, and so on. So, as you can imagine, there is a lot of an extensive engineering and scientific work which is performed all the time within EDF on assessment of risk, safety, and so on, because that's the priority of the company. But still, my expose today is not going to be about that. It's going to be our environmental impacts and all the R&D we are performing to mitigate such impacts. It's working. About mitigation. So first, about carbon emissions. So we EDF took the commitment to tend with carbon neutrality at the horizon of 2050. So it means that we have both to reduce our emissions, so to further increase the proportion of low carbon energy sources within our energy mix, and also to study and implement efficient, scientifically based carbon compensation or sequestration solutions, because still we're gonna have a residual carbon production. About biodiversity, we promote a positive biodiversity conservation initiative, and of course we do everything we can to avoid, minimize, and mitigate our residual impact on biodiversity. To do so, we have to first, sorry, I'm lost in my notes. Yes. We have first to objectively and accurately identify and quantify those impacts. We have to take into account global changes in both this evaluation of impact and also in the selection of the most effective over the long term, because things are going to change, obviously, mitigation solutions. And current approaches and regulation are mostly based on classical approaches focused on species and focused on, conservation, species that are concerned with conservation issues. But however, we tend to use as much as we can more global and integrative approach, and that's where the link is with the subject of our conference today and the notion of function. The last part of my expose will be about the R&D we are performing within EDF, because as you have understand yet, thanks to the colleagues who have highlighted our direct contributions from the scientific point of view, we are not only funders here, we are also we have also a team of researchers that are trying to do actual science, and that's the point of the last part of my expose. So very quickly, because it's not very interesting, but EDF R&D is yeah, it's quite a big machine with about 2,000 people involved. Among them, there is a little bit more than 100 of researchers that are dedicated within the Laboratoire National d'Hydraulique and d'Environnement to studying, they are basically working of all the issues I've presented before from both the angle of the environmental aggressions and the environment protection. So a few words now about the Laboratoire National d'Hydraulique et d'Environnement, which is the department that house our team and our skills. It's composed of 5 different teams. 2 of them on the left are more dedicated to hydraulics, combining a complementary approach using state-of-the-art numerical simulation tools that we are developing within EDF R&D, and physical model and mock-ups of different sites, as you can see here on the pictures on the left. We also have a team dedicated to water quality, hydrology, hydrogeology, and radioecology. Another one which is studying microbiology and environmental chemistry. And last but not least, because that's the one I'm managing, the team which is dedicated to ecology, ecotoxicology, and also which is supported by a little team which is performing in situ measurements for all of our departments and also for the ecological purposes. We also have 3 common research teams within that department. One with École des Ponts et Chaussées, which is called Laboratoire d'Hydraulique Saint-Venant, dedicated, named to study hydraulics. Another one with INRAE, dedicated to hydroecology, and the last one, Mélusine, is with the LRGP, it's Laboratoire de Réaction et Génie des Procédés in Nancy. To finish my presentation, I would like to focus on what's most interesting for us today, and is to give you a few examples of some research we are performing on ecology within EDF R&D, within my team. We are basically working for every kind of type of electricity production facilities, a lot, and historically a lot for hydroelectricity. We are studying, as an example, the impact of the variation of flow rates and water heights due to dam operations. On aquatic ecosystems, we are especially focusing on migratory species which are protected, and so we are working on their behavior and population. We are developing innovative monitoring tools, environmental monitoring tools, and solutions that can be implemented to ensure connectivity, meaning to prevent a dam from blocking them from migration. About nuclear, the example I gave here is that we have already, and we are launching a new co-constructed, by co-constructed I mean with a lot of different partners, external partners, research partners. So we are working on the construction of a new R&D program whose objective is to better understand the impact of thermal releases in the environment in a climate change context. About terrestrial ecology, our topic of interest are soil functions, land artificialization, carbon sequestration. We are working also on population and habitat connectivity models, and we are developing with partners. This example is with both Muséum National d'Histoire Naturelle and INRAE, tools to evaluate ecological quality of mitigation solutions. A new subject that is growing within our team is about renewables energy. We are currently developing new skills to better understand renewables energy environmental impacts. And to conclude this expose, I just want to highlight the fact that it's extremely important for us to work with partners. So you have some of them here. It's basically our main partners, but it's not a complete list. For 2 reasons. First, because as you can see, there's a lot of different topics. In our team, we are about 20 to 30, but only 15 people are really dedicated 100% to ecology. So we cannot be experts on all that topics. So we need to get some external expertise when we want to address a new subject. So that's the first reason. And the last reason is that if a company like us come and says, don't worry, I study something and I promise you we have no impact, you understand that it's not enough for the regulators, it's not enough for the public or for the scientific community. So we need for our credibility to be partners with the scientists, objective scientists, which are not part of EDF, to show that our work is our scientific work is of good quality, and that we are not lying to the regulators just to can do whatever we like. Well, it concludes my expose. So thank you very much for having us here. Again, thank you to the organizers. Sorry, I wish you all very interesting seminar this afternoon, and thank you again.
Okay, so good afternoon again. So now I would like to make some arguments about a possible shift of paradigm in conservation biology from species to functions. So the idea is to see whether and how we can see conservation of biodiversity through a functional lens. So there are many visions of conservation. And if I were caricatural, I would say that on one side, we have SOUBELET, who define conservation as the science of scarcity. So the focus is clearly on the protection of species. And on the other side, we have an ecosystem level focus of conservation, especially, for instance, by Carrera, who said that people need nature, and saving natural capital is an investment in our future. Of course, this last vision participates to the development of ecosystem services. With maybe a paroxysm is a kind of monetization of nature, in which you could put a price on for any kind of ecosystem service. As you can imagine, this leads to a lot of criticisms. For instance, Silverton criticized the neoliberalism and monetization and anthropocentrism of this vision, ecosystem visions of nature. But more broadly, several conservation people, for instance, Goldstein, criticize the fact that the ecosystem level visions of conservation, especially because ecosystem services, even biodiversity and the link between them are kind of buzzwords that are too vague for conservation people, and that we should still focus on the protection of species. However, it's clear, at least has been discussed a lot, that there is links between biodiversity, the structure and functions of ecosystems, and those functions are the biological basis of ecosystem services. But it's also true that from a conservation point of view, there is clearly different visions, that protected areas and conservation status mostly focus on the conservation of biodiversity and species. And on the other hand, we have global initiatives like the Millennium Ecosystem Assessments that focus on protection, at least assessment, but even maximization of ecosystem services. Of course, there are intermediate situations. For instance, in marine ecosystems, so protected area have been designed at some point to maximize fish biomass for fishing, which is a function of the ecosystem. But these questions of the linkage between biodiversity and ecosystem functioning is not new at all, for sure. And I guess you you all know in this room the early work in the 90s and later about the linkage between biodiversity and ecosystem functioning, especially ecosystem productivity, both theoretically and experimentally. For instance, probably know this well-known figure in which on the x-axis you have the number of species, on the y-axis you have ecosystem productivity, with a positive relationship, meaning that the more species, the more productive an ecosystem. This is a kind of ecology by numbers because we don't care about the identity of species or the role of species. Just if you have one more species, you will increase your ecosystem function. But it's a bit more complex, in fact. In fact, for instance, when you look at these different communities of plants or fish, coral reef fish, well, there are differences. This guy is really big. There are some smaller organisms. You have plants with big leaves, plants with small leaves, and so on. So maybe they should play a role in the ecosystems. And this is the focus of a discipline of ecology called functional ecology that use different tools to understand the role of individuals and species in communities and beyond by measuring features on organisms. Those features are called functional traits. So that can be morphological, but not only. It could be the physiology, the phenology, or even the behavior of organisms. It is really useful to understand the role of species in communities. So we have a non-exhaustive list of functional traits for different taxonomic groups. For instance, for plants, seed mass, plant height, or rooting depth can be related to different functions the plant can display in an ecosystem. So functional ecology focuses on phenotypes of organisms, but what is interesting is that, in fact, there is a change, I think, of the representation of the phenotype over time in functional ecology. At the early beginning, functional ecologists really focus on individuals and really look at individual measurement. It was a really mechanistic point of view to try to map the structure to function, to understand the structure to function relationship. But then over time, because maybe a need of generalization, predictions, we lost a bit this individual approach to focus on species average and to compare species across ecosystems, across biomes, and so on. And even more recently, now the community is to go on, the exercise is to go on this aggregation exercise at the community level by focusing on phenotypic average values at the community level, which can be kind of community phenotype, which could be really useful to understand and predict the response of communities to environmental change. And the paroxysm is maybe has been reached something like 10 years ago with the rise of big data, big database, and large-scale analysis, and so that we can tackle classical biogeographical questions through a functional lens. But in the meantime, there has been a return of interest to look at intraspecific variation, especially to try to bridge ecological and evolutionary questions. And more recently, also, we start to be interested by some extreme phenotypes in populations and communities and why they are maintained. So indeed, in a population, in a community, we can describe trait variation, phenotypic variations by mean, variance. But this guy could be interesting too. Is it an outlier or not? Is it important to account for instance, for ecosystem functioning? So when you look at this community, you can have different species with more or less individuals, but also with different phenotypic values, some organism seems to be different from others. This is what we call the ecology of outliers, and our idea is to see whether and how these outliers could play a role across scale and time. So how to define those outliers? Well, first, I think we all acknowledge that the phenotype is quite complex, so it's difficult to summarize it only by a single traits. But it's quite important to look at several traits simultaneously to account for the fact that the phenotype is multifaceted. You can quantify this, for instance, with multivariate analysis like PCA. And then the idea is to see whether and which species or individuals are outliers. So for that, we develop different indices, like the functional distinctiveness, which is quite easy mathematically. This is just the sum of these distances from all others. So we even developed a package as part of our CESAB working group to help characterize these outliers. But more interestingly, it's really interesting to see why we still have some outliers in communities. So what we start to do is try to apply first principle from evolutionary biology by looking at kind of fitness landscape, but not of a population, but of a community, so across species, to see whether and how we can explain why the maintenance of rare phenotypes in communities. And one reason, among others, is that we could have local fitness peaks, so that explain that, yes, they could be adapted to some specific abiotic conditions or species interactions or natural processes. Okay, so we have this kind of new indices, functional distinctiveness. What does it mean compared to more classical view of rarity? So if we look at this big plot to put two kinds of rarity. On the x-axis, we have abundance scarcity. So it's a classical view of rarity, like Soubelet proposed, just if a species is abundant or not at local or global scale. You can be dominant or scarce. But you can also have traits that are really ordinary, common, redundant, or traits that are really distinct from others in the community. So we have another axis. So in fact, we can have all these cases with, for instance, species that are dominant with distinct traits, or species that are scarce, but with redundant trait values. Of course, we can imagine that the things to conserve is here because the species can be scarce and with distinct trait values. Okay, but does it mean that we have no relationship or not? At least we expect no relationship between these two facets of diversity. So we test that for different data sets. For instance, for global reef fishes. So each dot is one species. On the x-axis is just abundance scarcity. And on the y-axis is functional distinctiveness. And indeed, it seems that there is no relationship at all between these two facets of diversity, which means that maybe, yes, it could be important to account for both facets of rarity in conservation biology. But why? Why it could be important to consider both facets? Maybe because it's important for ecosystem functioning. So these questions, it's not so easy to test, to be honest, especially experimentally. So one of the ideas is that now we go back to plants, trees in particular. So think about 6 tree species. 4 have really common redundant traits, and 2 are extreme values, have distinct trait values. So we can expect that if we are losing species, so if there is a biodiversity erosion scenarios, by random, we have this kind of relationship. But if we are losing the most distinct species first, maybe the impact on ecosystem functioning, like productivity, will be higher and faster because we are losing some ecological niches. On the opposite, if the species is redundant, it's a pity for sure, but maybe the impact won't be so huge. So to test that, again, it's not so easy to test with experiments. So what we did is in silico experiments with modeling. So for that, we used a mechanistic model of three ecosystem dynamics and functioning. So for that, we used the forest gap model for Cefe, developed by Xavier Morin. And what we did is that we tried to test that with different sets of scenario simulations by removing one species, then another from 0 to 30, with simulations in which we remove, so we have thousands of simulations. We remove all species totally randomly. And for others, we have some rules where we remove the most redundant species first in blue, or the most distinct species first in red. And here is one of the results. So on the x-axis, this is the number of species we removed in those simulations from 0 to 30. And on the y-axis, it's the total biomass of the forest, of the simulated forest. In gray, this is all the simulations, so hundreds in that case, where we remove species randomly. In blue, this is when we remove one by one the most redundant species. And in red, this is when we remove the most distinct species first. And in fact, we exactly found what we expected is that indeed, when we are losing the most distinct species first, we have a drastic decrease in ecosystem productivity. What is interesting is that we run these models in different climatic situations, and that we found this especially in the most extreme environmental situations. So to conclude on these parts, it seems that, yes, beyond classical diversity effects, it seems that ecosystem functions can be regulated by the effect of some key species too. We try to look at this in empirical data with a big database of alpine plant communities for more than 20,000 plots. And we indeed found several situations where this is the effect of key species that matters more than diversity. Okay, so based on that, I would try to reconcile different conservation targets. I mean, on one hand, species, and on the other hand, ecosystem functions, properties, services, nature, contribution to people. So we make the argument that it could be good to still focus on species, not diversity per se or not ecosystems, but on functional distinctiveness of species. That could be the missing link between these different conservation targets. In that case, it's important to look at the contribution of those species to diversity. Functional distinctiveness per se, if you are really distinct, you are really important for the whole biodiversity and trade diversity in particular, like sharks or salmon. But in the meantime, to consider the contribution of those species to different ecosystem functions. If we think about ecosystem productivity, well, tuna or salmon can contribute more to productivity than the two others. So we could try, and this is what we are doing now, try to do this exercise for a lot of different taxonomic groups, for instance, fishes, plants, birds, to classify them, the species, in terms of functional distinctiveness, if they are more or less distinct, but in the meantime, to consider their contribution to ecosystem functions. So first, for instance, productivity, but also other ecosystem services like cultural values. This could be the case for pollination and others for sure. And as you can see on this graph, of course, is that because of biological trade-offs and so on, while a species is not doing a great job for all this ecosystem service, but this is the start of optimization exercise we start to do to see whether and how we can account for both functional distinctiveness and different ecosystem services in a more functional view of conservation. So to conclude, we are wondering if we are starting to have an emerging functional conservations, especially because early work in conservation biology have been developed in kind of static world. So the idea was to predict the exclusion of species in response to land use. But now, because the world is changing, it's more difficult to predict which will be the losers and the winners of the global change race. So in that case, functional traits can inform the conservation status of species and help predictions. We have done this exercise about the conservation status for many taxonomic groups. Again, here I'll go back to coral reef fishes. So on the x-axis, it's the different classical IUCN status, threatened, not threatened, data, no data, or not evaluated. And what we see, in fact, is that in the big group of species for which that has not been yet evaluated for conservation status, this seems to be quite distinct from others. So for sure, it could help. It could also help a triage exercise for conservation planning and for assessing the different species on Earth. And so functional distinctiveness really of species, not diversity per se, can be helpful to inform both the contribution of species to diversity per se, but also to different ecosystem functions and services. At least this is our bet. So I thank you a lot. And again, I would like to thank you a lot, FRB and EDF for all this work. And all the members, there are a lot of my free working group, there are more than 40, I think, that contribute to this talk. Thanks a lot.
Thank you very much. It's a very interesting presentation. I'm just very curious of detail. How do you measure the productivity, the contribution of one species to productivity?
So for ecosystem productivity, it's ecosystem biomass. It's not so difficult. It's relative biomass production of each species, but we assess it also statistically with different statistical models, a pipeline of analysis in which we look at the effect of diversity per se on ecosystem functions. And then we add one by one the species and we compare different AEC values. And so we have a set of species that significantly contribute to ecosystem productivity beyond diversity.
Yes, thank you, Cyrille. It's a little bit of a follow-up on this question. How do you account for species abundance in your productivity and the functional rarity relationship?
Thanks. So, well, of course, we analyze both for sure. And the idea is to decouple to see whether independently of biomass and species abundance, there is an effect of distinctiveness. So in the model, in the statistic models, we account for both. And in some situations, even for rare species, for scarce species, we have an effect of distinctiveness. So we did both. I mean, in our analysis, we account or not for species abundance to see whether and how functional distinctiveness matters. Sorry? But for instance, in extreme environment, yes. And the next step is to also test that for the stability, resilience, long-term productivity, for which we expect also a strong impact of functional distinctiveness on the long term, even if those species are really scarce.
Thanks a lot for the intriguing talk. Hey, I was wondering about the conservation value of functionally unique species. So imagine the hummingbirds. So there are hundreds of hummingbirds, but they all look very much the same to me from a distance. So are you saying then that we save one hummingbird, that's enough, and then better go to another clade and save another functionally distinct species? Or are we fascinated by this tremendous radiation of functionally quite similar species?
So it's here, hummingbirds. Huh? so our framework is flexible enough to apply it at the species level or at a group level, genus level, family level, and so on. So one way to do that is to focus on genus or something like that and say that, okay, at the genus level, which genus is distinct or not. This is something we can do for sure. Practically also, we have different metrics that account for uniqueness. In that case, this is the fact that indeed one hummingbird could be replaced by another. But if one hummingbird is distinct with functional distinctness, another will be two, because mathematically this is the sum of distance to all others. So in that case, the idea would be to conserve and protect all hummingbirds. Yeah. Again, I think this framework is flexible enough to apply it at different spatial scale, and I think that one of the byproduct of all these questions is that conservation is context dependent, and that we should assess the functional distinctiveness and the contribution to ecosystem services and functions at local scales. So in that case, yes, a tree can be really important in grasslands at local scales. So it's also important to look at it at local scales and not only at global scales. Right now, what I see when talking about functional trait diversity is, for instance, for me, it's the end of the gradient. It's, for instance, it's essential variables of biodiversity that it's at global scale. You put traits because you have the data. For me, it means nothing. It depends on the questions, it depends on the objectives, it depends on the context. I think also another message is to go back to local.
I wasn't going to ask about the same question, but in some, what you're saying is that you're using that as a heuristic, in fact. It's not, it's something you should consider in general and go back to the particular situation. Okay, well, why not? I don't know whether it's very practical, but we'll see. So the point is also whether, I mean, the world is changing very fast, so the landscape also. So how do you account for that? I mean, something that is useful because it's rare or interesting at some point, and then the temperature is growing by 2 degrees. What do you do as an advice for conservation biologists? Well,
theoretically, clearly, we expect that the functional distinct species today will be really important for resilience of ecosystems. So I would actually suggest to protect them anyway, because they can play a role in the resilience of ecosystems and in the functioning on the long term. So for sure. And, more broadly, regarding your questions about the fact that the world is changing, at least when we build some trait environment relationships, we can provide some generalizations for conservations and rules to help them target the species that maybe will extinct faster than expected. I cannot answer these questions now. It really depends on the questions. For me, this is one of the messages. It's why it's hard for me to understand putting threats in conservation program without specific questions in mind or specific objectives, for me, means nothing. Huh? Except on one aspect is that in a multivariate perspective, so we did this exercise with different taxonomic groups, and when we have at least 5 or 6 threats, irrespective of which they are, they capture a lot of the functional space of species. So if you really don't know, at least use a lot of threats, they should capture something. This is what we found in our analytical approach. Even, to be honest, I'm not a big fan of this. I would prefer to have specific questions in mind to use threats.
So related to the question on abundance, but regarding the result of Xavier Morin model, how do you control for like the contribution of each species on biomass and making sure that the distinctiveness, the functional distinctiveness, like the rare species, the rare function, are not the one contributing the most to total biomass?
We cannot control for it at the end, but we control for it at the beginning. Because at the beginning, all the species have the same species abundance. At the end, for sure, the most productive one in some environment was those with the more individuals. But at the beginning, all species have the same abundances.
And the same biomass?
So we start from nothing, so yeah, I would say that.
Thank you, Cyrille. I think we have to move forward, so you can keep your question for the coffee break. Thank you.
Yeah, so it's still me. I'm really happy to introduce Ana Rodriguez. So she's a senior researcher at Cefe, and she has long worked on conservation questions at local and global scales. So thank you.
Thank you. Just check it works. Thank you very much, first of all, for the invitation to this conference. It's a great opportunity for me to discuss some ideas that I've been fermenting in my head for a while, and it's great to be able to discuss them with you. So I'm a conservation biologist. I come from the conservation side of things. I am not a functional ecologist, as you will find soon by all the I will say for sure. So I'm coming from that perspective. I'm coming from the perspective of trying to understand, seeing a functional approach to conservation, coming into conservation biology and thinking, is this useful? Is this something that we can use in practical conservation? So like Cyrille, I'll start with a photo of an old man. In this case, Aldo Leopold. He has this, so it's an influential thinker into environmental ethics. And there's this famous sentence that he has: to keep every cog and wheel is the first precaution of intelligent tinkering. And this has been a sentence that is very seldom, very, very commonly used to justify the conservation of species. I put, what's before that. The last word in ignorance is the man or woman who says of an animal or plant, what good is it? If the land mechanism as a whole is good, then every part is good, whether we understand it or not. If the biota in the course of eons has built something we like but do not understand, then who but a fool would discard seemingly useless parts? So this is the precautionary principle in action. Even if we don't know what it's for, let's just not get rid of it. And lots of conservation, lots of the effort around conservation of species has this explicitly or implicitly in heart, this idea that we should keep all the parts just in case. Because they might come useful or because they're good in themselves. So avoiding species extinctions, for example, is a major drive in conservation planning. But he also mentions the land mechanism, and I'll be using this metaphor a lot in the talk. It could be said that actually we don't just want to keep the parts, we want to have the functional mechanism, we want to have the full thing working together. We don't just want each species in zoos. We want the species interacting and doing what they do, having the land mechanism together as a whole. And it could be argued that this is the change of paradigm that maybe this conference is trying to address. It is the promise of conservation, a functional lens to conservation biology, is that allows us to go from a perspective of keeping the parts to actually conserving the whole, conserving the full mechanism. Now, there is a lot of research, of which I have not participated at all, but several people in this room have made major contributions on the relationship between biodiversity and ecosystem functioning. And I will not go into that, but I'll just take these as truths, that this research has found that usually when there's higher diversity, and specifically higher functional diversity, there are more efficient and stable ecosystem functioning, usually efficiency measured as productivity, and stronger provision of ecosystem services. Let's assume this is true. So I wasn't sure if this was going to be explained before. Cyrille already mentioned this idea of functional diversity. What is this functional diversity? So if you think of each species as traits, so in a multidimensional, hyperdimensional trait space, each species has a position. If you have a set of species which are very spread, you have high functional diversity. If they're all very similar to each other functionally, you have a small trait space that they occupy. And so this is how you measure the functional diversity, correct me if I'm wrong, the functional diversity of a community. So this higher, when I speak about higher functional diversity, usually it means these, although they have many metrics and they're very complicated, but usually kind of this is the general idea, at least the one I have in mind. So this is the research has shown this. And so the promise of a functional approach to conservation is that if we conserve functional diversity, we will be conserving ecosystem function and the services that we derive from them. So my talk here is coming from the conservation side and is asking, is the functional approach to biodiversity conservation informing conservation priorities? And specifically, because that's what I work with, I'm interested about spatial priorities. So areas which are priorities for conservation, for example, for the designation of protected areas. And arguably, protected areas are precisely the place where we want whole functioning things, right? Because that, by definition, they are ecosystems. When you're thinking about species-based conservation, you might be protecting one species at a time. But when you think about protected areas, that's where you want functioning wholes, not wholes, the full mechanism working. So there's a big literature on conservation planning on how to select priority areas for conservation. And when the functional approach came when the functional approach arrived into conservation planning, it came into these kind of studies. There's several of them. This is one of the landmark ones, the first ones. This idea that if you map, you can map different facets of biodiversity. So you can map taxonomic diversity, for example, species richness. That's not exactly what it said, but something that represents taxonomic diversity. You can map phylogenetic diversity, which I won't be talking about here today. And we can map functional diversity, as I mentioned before, this idea of the diversity of the community in terms of its functional traits. There's lots of studies that have done this, picked data and looked at these three facets, taxonomic, phylogenetic, and functional diversity. And the result is pretty universally the same. There's some correlation, it's not perfect. There's always places which are important for some facets, but not for others. The conclusion being we have to keep all facets in mind because they are representing different things, different aspects of conservation. So the priorities should come from these two facets coming together. So, and this is presented, adding the functional diversity facet is presented in the context of saying, if we do that, we will be accounting for ecosystem services and processes. So for example, this is a quote, this is a classical quote from these kind of papers. Functional diversity enhances ecosystem functions such as productivity, resilience to perturbations or invasion, regulation of water matter, therefore critical to know how it's distributed across the globe if we're aiming for efficiently protect communities and ecosystems beyond species. So this is the argument used for this kind of analysis. Now, when I see this coming, I think, okay, it's anchored in this research it's anchored in this research that has shown that functional diversity does contribute to stable, efficient, and productive systems. But this research is done at local scale. This system picks as a mechanism, and as Cyrille has just mentioned, you can pick some species either mechanistically, or experimentally or theoretically, but you're playing with the system, you're playing with the watch, you're playing with the clock, you're taking things out and seeing how it works. So by all means, I think this research is extremely useful to understand this, to help us understand what happens when a given ecosystem changes. So for example, a study showed that in the Hawaiian Islands, the net effect between extinctions and introduction of species richness has more than doubled, but yet functional richness has declined by 70%. This is interesting. This is telling us absolutely functional species diversity is not capturing everything. This is telling us information about how we need to manage and to protect these islands. But when you think about this at the global scale, I'm not sure anymore what this research is helping us define priorities at the global scale, because suddenly we're comparing different mechanisms, and I am not convinced, this is just my point, I am not convinced that this research means that therefore this area is a priority for global conservation, because if we protect the areas more in red there, we'll be missing on all the other mechanisms. And it doesn't necessarily tell me that the global system, the global functioning has improved. So I think there's a mismatch here between scales of where the evidence comes from and how the argument is being used. Which brings us to functional rarity. So that's, we're in the hotspot of rarity itself. If I say something not correct, feel free to correct me. So this is one of the major outputs of the free group, and this mapping of species which are rare on two axes. So they're both geographically restrictive, so it's small ranges, and functionally distinctive, exactly what Cyrille has just presented to us today. And so if rare species are not redundant with other species and instead hold unique combinations of traits, they will contribute disproportionately to ecosystem functioning and associated services. We've heard about the evidence towards that, and let's assume this is a true statement. Let's assume this is correct. Example of cuckoo watch. If you miss the cuckoo in the cuckoo watch, it's a very important part of it. You don't want to miss it. But then how this translates into global priorities is where I have more trouble with, because so then the argument, given the large number of ecologically rare species currently threatened and experience population declines, the biosphere will be facing a major functional extinction crisis if effective target conservations are not implemented. So the authors argue for these being a priority area for conservation of a priority based on functional arguments. To me, this is an argument coming from the keeping the parts. Yes, let's protect that for sure. But let's protect it because these species are very unique and very strange and very bizarre, and they're doing different things. I don't think we have an argument for saying that protecting that is going to protect the function at the global scale. Actually, I would make almost the opposite argument, that the species that we really need to be caring about at the global scale to protect the function at the global scale is the widespread, functionally distinctive ones. So the ones that are in many places, but which are distinctive and which are threatened. And those species, if they disappear, a lot of systems are affected at the same time. And we know some of these species already. Big predators, for example. Functionally, the biggest animals, they're not very common. They'll probably come up as functionally distinctive, I don't know. But they are widespread, and their disappearance is indeed having a major effect on ecosystems around the world. These species, their disappearance will have an effect there, but only there. They will not have an effect where they're not, by definition. So I'll just use a parallel now with the now into the phylogenetic side of things. The EDGE program is a program that it's kind of conceptually similar to these functionally distinctive species. It looks at species which are evolutionarily distinct. meaning, so you have some species that have lots of related species, that there's lots of rats. They all look more or less the same, you lose one, no problem. But then you have things really weird like these ones, like the platypus, which if it disappears, there's nothing else like it, it just goes evolutionarily. So the EDGE program has identified as a species distinctive, phylogenetic distinctiveness, evolutionary distinctiveness, and then looked among those, the ones that are more threatened. And the argument is, so it's if this species goes, the tree of life gets poorer. The way they describe it is the Hedge of Existence program protects some of the most unique species on the planet on the verge of extinction. They're weird, they're wonderful, they represent unique, irreplaceable part of our world's biodiversity. They don't even try to say that it's going to save the functional or ecosystem services. Same in the original paper that published these. The point being, if we lose these species, we lose a big part of the tree of life. That is an argument in its own right. It's a good argument. It's a good argument. And so I kind of think that the functionally distinctive species are a bit on the same page. These are going to be weird species. They're going to be interesting species. I don't know if the argument that they contribute disproportionately for a global function is that solid. Just final part. So in conservation planning, looking at hotspots of richness or rarity is very 1990s. Nobody does that anymore. People for past 20 years look at this idea of complementarity. The idea that you don't just pick the high areas of a given facet, but you look for networks of protected areas that together have a lot of diversity. So, for example, so this is diversity of mammals across the world, and what a complementary set for the protection of mammals could look like would be something like this. So you're seeing that if you just use richness, you would just be picking tropical areas, while this one is actually picking areas in all ecosystems. So it's making sure that all species get represented somewhere. So this is the standard in conservation planning for highlighting priorities for protected areas for a long time. And people have applied that. And this is a completely pick the parts approach. It's a save all parts approach. Make sure that protected areas have all the parts, which in this case are the species. And the same logic exactly can be applied to if the parts are traits or if the parts are phylogenetic diversity. So this paper did just that. You looked at complementary based planning on the taxonomic dimension, on the phylogenetic dimension, and on the trait dimension, making sure that you have a complementary set of traits. And then the overlap, the green areas are good for all three of them. And I'm fine with these, but it's a completely save the parts approach. And yet, that's not how it's sold. The way it's sold is it's crucial to ensure the provision of ecosystem services and the contribution to human well-being. I don't think that by saving one trait from one ecosystem and a trait from another one, even if they're complementary, they're not together. They're not functioning. So I'm pretty much done. But just give one good example of something that actually I think is a good use, a functional approach. It's less sexy, but I think actually it's making much more of a difference for conservation. The key biodiversity areas approach. So for key biodiversity areas, which are defined as sites contributing significantly to global persistence of biodiversity, it's very, very much bottom up. It's you don't need data on the entire planet to find out what's important. You just need to see if your site meets the criteria. And there's a set of criteria, and some come from the save the parts, and some come from the save the functions. So for example, if a site has threatened species, which is clearly a save the parts approach. If it has rare species or rare ecosystems or rare assemblages, save those parts, make sure that you keep them. But then it has sites of ecological integrity. These things are still being developed. These actually the criteria, the specific way these criteria are being used is still in progress now. But for example, for the ecological integrity criterion, what you want is for each ecoregion up to two, the most intact sites, the best sites, the sites that have the most intact ecosystems. This is a great place to use functional diversity. This is a place to compare between sites which are otherwise relatively similar, but the ones which are in better shape. And another one is criteria that look directly at biological processes. So for example, places of aggregations. So this is to protect the process of migration. So if you have places where species, particular populations concentrate particular times of the year, that's important to keep them, because otherwise you will lose the process of migration. Ecological refugia is, for example, sites which are important, but not always. For example, there might be a wetland that becomes really important in dry years, while it's not very important in usual years. And recruitment sources, which is very tailored to aquatic systems, this idea of places which are very important, nurseries, places that produce propagules and juveniles and so on. So for me, this is an excellent example of how you can unashamedly have both keep the parts and save the process, save the functioning system. So to finalize, I think the functional approach to conservation is really interesting and really useful. I think they're not mutually exclusive, that we want to keep all the parts and conserve the functioning parts. I just urge everybody to have a better alignment between the arguments that are being used to justify the conservation approaches and the approaches. Thank you very much.
Thank you very much. That was very, very interesting. When it comes to ecology, I'm used to say that we are living the golden age, of ecology. I mean, so much people, data, and concepts all together, facing a crisis and so on. But in terms of conservation biology, sometimes I feel like we are still in the stone age, because we are disconnected from the mechanisms that lead to the assembly of the ecosystems we'd like to conserve. And so I would say that the functional approach is very reductionist, and it's making a lot of mistakes on its way. I would say that the what is not that constrained. That example is very good, but you take a very constrained ecosystem. Ecosystems are not that constrained. So, but anyway, but I would ask you how an effective conservation biology would be if it would not be mechanistic. What would be the alternative?
But you have to be mechanistic you have to. But the aim of conservation comes before that. The why you're doing it? What's the purpose? It's like, what do you want from conservation? What's the purpose? And that's a moral issue. There's a part which is also we won't survive. We want the planet not to go. But there is a part which comes then to, is it because of existence value? Is it because of insurance value? All of these things come before, and those are a society decision. And the decision to keep all the parts, it's embedded. I mean, it's embedded that you use it. you say this species, we should not be losing this. These are unique. We should not be losing this. The part of saying we also want this function, the function ecosystem, it's necessary. If you want to protect a species, you have to protect it in a functioning ecosystem. So absolutely, you cannot ignore it. But the decision of what you want to protect comes before.
Yeah, but we're going to have plenty of time to discuss about that. But still, I think it's not a question of what. I mean, it's a question of we are in a crisis right now. The other decisions are made. And for instance, I mean, you take that example of the very restricted and functionally distinct birds that might not be that much important, at the global scale. And you say that, and you're right, that the widespread specialists might be more important, but those species might be not in danger of extinction. But the other one, the restricted, functionally distinct, they are.
Okay, but so I said at the same level of threat and the same level of functional distinctiveness.
Yeah, but they are not, by definition, because they're
geographic. You're losing lots of top predators.
Yeah, you're right. But we'll talk about numbers afterwards. Thank you very much, Anna.
No, thank you.
Thank you very much for a wonderful talk. It was really fun to think about. So I just want to, I'm a community functional ecologist, so I'm not a conservation biologist. So please, if I get out of my zone, please put me back in it. But in terms of the watch metaphor, I'm trying to think about what Aldo or others would think about if they, I don't know Sand County Almanac that well anymore. It's been a while since I read it, but the pieces of the watch are moving across the landscape in idiosyncratic ways, right? We know that from paleoecology. The watches don't move together on the landscape. which means that we're always getting new watches. I mean, the watch is always changing, like regardless of what we do. So I guess I'm just trying to think about how you incorporate that dynamism and the fact that the pieces move in different ways, in idiosyncratic ways. We've known that since Gleason, right? So how do you account for that dynamism in the kind of save all the pieces framework? I'm just trying to kind of make sense of that, I guess.
Well, the watch for me serves more as a way of, so there's two metaphors here. One is between separate bits working together. So then I made them in. And then the other way, the way I use it is the difference between looking at global arguments versus communities. So that's how I use it. Of course, communities are not isolated. So that's just one part of, it's a metaphor, like all metaphors, there's limitations. How do you account for resilience? You're saying keep the pieces, but in a resilient way. You can decide first that you want to keep all the pieces, like you want no species to go extinct. So your priorities become the species which are more threatened. And then you think, what do the species need to be resilient? So it can go in that direction. It's not, these are not mutually exclusive objectives. So for example, I gave the key biodiversity area approach. It looks at places with threatened species. It looks at places with rare species. So try, even if they're not threatened. So saying this, we should keep an eye on this in case this becomes a problem. It looks at, for example, conserving processes such as migration, even if lots of migratory species are extremely abundant, they're not threatened, but keep an eye on them because they do go through bottlenecks in time and space that if you don't keep an eye on, we'll lose these processes. So I think you can do it more explicitly. Say what you want. You want to protect migration? This is what you need. You want to protect productivity? Because a lot of the discussion between the relationship between diversity and function is productivity. Do we want to protect global productivity? Is that what we're talking about? Sure. But we as humans are very good at manipulating productivity. So it would not be my first priority in terms of global targets.
Thank you very much.
Thank you.
Thanks a lot, Anna, because you put the end on something that I've been burning in my head since 2 years about the functional distinctiveness. And I agree 100% with sometimes the mismatch that we can see between the argument of functional distinctiveness is good for ecosystem functioning or productivity. And there is a mismatch between this claim and thinking of functional distinctiveness at global scale. But I think it can be a limit of how the concept is used, but not a limit of the concept, because once you compute this functional distinctiveness inside between the species where the ecosystems is actually functioning, for example, the community or the species assemblage, local, then the argument, you can make this argument of the link between functional distinctiveness and the ecosystem productivity. And in this sense, the concept is working, I think, more for this claim.
But I didn't argue, I mean, there is some evidence that functional distinctiveness does contribute to disproportionately to the functioning of the community. But it can only do that for the communities where the species occurs. So the question is, it then does not give you an argument that globally it's a priority. It tells you that's a priority there.
Yeah. But you could think of looking at this local functional distinctiveness at a global scale.
You could, but
we go back to the importance, I think, of the context dependence that you said.
There could conceptually be rare, restricted in the distribution and functionally distinct species, which only occur in a given site, which are globally very important. But that's not what the literature has been working on. The literature has been working on the local relationships. There could be, I don't know, there could be a species which is functionally distinctive, which occurs just in a place, and it's the cure for cancer, as an example. This species might well be globally important for ecosystem services, human health. There's an example. But that's not what the literature has been working on. It's not been trying to understand if those species which are ecologically distinctive are globally important.
Yeah, I follow you.
Sorry, I talked too much.
Yeah, thank you, Anna, for your presentation. I'm now curious about what do you think is the reason to preserve biodiversity, actually, as a conservation biologist?
I think all arguments are good. I just think there's no shame in also assuming we want to keep the things because they're unique and weird and different. There's no shame in that, in having that. We also want functional ecosystems for sure. So for me, the arguments, they come from all lines of intrinsic to insurance value to applied value. All those values are good as far as I can tell. But I have no problem with also because they're cool and they're very different and there's nothing like that.
Okay, thank you.
Now, before the break, I'm really happy to introduce Brian and Chris, who is a professor at the University of Arizona and part of the free working group. Brian is well known for a lot of stuff, including macroecology, biogeography, and is the leader of one of the biggest plant database worldwide, the BN database.
Thank you, Cyrille. Thank you for the organizers of the conference. This is fantastic. I'm going to take a different approach, but I hope to land in between the two excellent talks that were just given. Biodiversity, right? That's why we're here. It's defined as the variety and variability of life, right? And so we get hung up on what we mean by variability, right? And so I think it's clear increasingly that we care about numbers, number of species, species richness, and so on. But we're here because we also care about what species do and the diversity of things that they do, the diversity of functions, diversity of traits, diversity of their impacts. So we're also here in the Anthropocene, right? And it's scary. And as biodiversity scientists, as a science, our main goal from society is to predict the future, to predict what's going to happen. So can we do that? Can we actually say, with the confidence of our atmospheric scientists, what's going to happen to the biosphere and what's going to happen to biodiversity? So here are our futures. We have various different futures ahead of us. And these futures are known as RCPs, or representative carbon pathways, or different climate change scenarios, where we have worst case scenarios, middle of the ground, or best case scenarios if we get our act together. And each of these different scenarios are going to have tremendous impacts on the biosphere, on the functioning of the biosphere, through their impacts of species and how species then impact various different ecosystem and biosphere processes. And so this upcoming planetary boundary that one hears in the literature has to do with this divergence of these differing scenarios. And these differing scenarios in terms of biodiversity are going to be expressed in terms of not only shifting species distributions, but mainly in terms of extinction and extinction risk. And so we talk about these critical thresholds of crossing then this planetary boundary. And the worry is that we're going to be entering increasingly into this sixth extinction. Remember, there's been various major extinction events on the planet having to do with the impact of asteroids and various other calamities on Earth. And now the calamity is us. So rapid changes in biodiversity are going to have cascading impacts on the biodiversity of the planet, but in particular, the functioning of the biosphere. So let's step back. So as scientists, in particular as botanists, we wanted to think about biodiversity of plants, an incredibly important group of organisms because they dominate the biomass on the planet, living biomass of the planet, but also dominate the productivity of the planet. And we wanted to ask some basic questions. In particular, how many plant species are there on Earth? Right? If we've got our act together, got all the data available, how many species actually are there? But we wanted to know what fraction of those species are rare. So we wanted to ask questions about rarity on a global scale, and then ask additional questions associated with species rarity. In particular, what's the future of rarity? We're interested in rarity because if we wanted to predict extinction, the best predictor of extinction risk is how many individuals you have. The rarer the species, the higher the probability or risk then of extinction. And so this talk today is mainly about rarity. And so the biology of rarity, but also biodiversity is mainly a story of rarity. Rarity, it turns out, may have a disproportionate, apologies for the PowerPoint failure here, impact on the biosphere. And that's what we're debating. Do rare species disproportionately matter? Okay. So the best predictors of extinction risk is small population size. So there are wonderful natural history and amazing scientists all over the planet trying to identify rare species. So this is Steve Perlman. He works mainly in the Hawaiian Islands, and he does these amazing excursions out. He rappels down cliffs, and he finds these extremely rare species, then of, in this case, orchids, identifying their populations, collecting seeds, collecting specimens to best preserve this rare species. Now, this is time-consuming, right? This work is extremely important, and we need to rapidly scale up and identify rare species and where they're located in order to best then anticipate extinction. All right, so we wanted to know, where are the hotspots of rarity, and is there anything special about these hotspots? So, can we measure the abundances of all species on Earth? Can you actually identify not only the common ones, but most importantly, the rare ones? Now, to do this, over the last approximate decade, we've been trying to compile and integrate the world's botanical data in a format which we can actually use and then ask these different questions. So we assemble herbarium data, vegetation survey data, plant traits, and other various different measures of plants across the world and integrate all these data together into something known as the BN database, or the Botanical Information and Ecology Network. And to give you kind of a short kind of overview then of the numbers, we have then compiled what we believe to be some of the better data sources where we have over 200 million observation records then of plants then across the world. And we can then use then this information then to come up with our best estimate of the total number of land plant species on Earth. And so we have come up with a number of about 435,000 species of land plants. Okay? An impressive number, and well within the realm of other expectations in terms of the total number of plants. But now, can we ask additional information about then species diversity of plants? And so we wanted to know, what is the relationship between the total abundance of species of all of these different species of plants then across the world? And so here we wanted to fill in this space, the relationship between the number of observations and the proportion of species. And so what does this distribution look like? So we have a question mark here. So we have some various different expectations. And so what we can do is we can go into the literature, we can go into various different ecological theories that we have to come up with an expectation for what this distribution actually looks like. And the first one actually comes from Steve Hubble's theory of neutrality, neutral theory. And what neutral theory tells us is that if we start with the assumption that in general ecological differences don't influence the abundances of species, that is, niche differences and functional differences then don't influence the abundance of species, to make a long story short, we should see a flavor of a distribution known as the log series distribution. That results in an inverse relationship between total number of observations and the proportion of species. In short, most species, according to neutrality, should be rare, and very few species should be abundant, an inverse relationship between abundance and the number of species. Now, we can mathematically express these distributions to show that the flavor of that distribution, if you plot it on logarithmic scales, it actually has a slope, a negative slope, And that slope should be around -1, it turns out, according to neutral expectations. And so it turns out that ecologically, we have a diversity of differing theories. Here's actually coming from niche theory that states that differences in abundances between species are due to ecological and functional differences. This one is known as the K-niche model. And in this case, we actually get a similar distribution. But the flavor of it, the functional relationship, turns out to be just a little bit different. Instead of a log series distribution, just like neutrality, we also expect a log series-like distribution as well, but the slope presumably will differ from -1. All right, so it's not going to be, it could be steeper or shallower, but presumably it wouldn't be -1. There's another prediction that also predicts a similar distribution. And this distribution, instead of being a log series, it tends out to be a Poisson log normal distribution. And this basically comes from the central limit theorem that states that the probability of abundance is set by a bunch of multiplicative factors. It could be the environment, other species, and so on, working together that influences how abundant then a given species are. And so presumably this functional relationship will also have a slope different from -1, but the distribution, the flavor of the distribution, is actually quite different. It's similar, but it's quite different. So what do we see? So we take all of this plant data from across the world and we plot the relationship between the number of observations and look at the proportion of species then that are characterized by a given kind of abundance class. And across these 200 million observation records and across well over 400,000 different species, what we actually find is that a functional relationship between observations and the proportion of species that is exactly characteristic of our third model, which is the central limit theorem. So neutrality, the K-niche model, don't fit the data, but instead the central limit theorem, which we all learn in basic statistics, appears to do the best job. Now what's interesting is if we look at the implications of this distribution, what this distribution actually tells us is that a large fraction, in fact, the majority of species on the planet are rare. And if we actually calculate the fraction of species then that are in the rarest categories, we actually find that of the well over 400,000 different plant species on Earth, approximately 36% of them have only been observed 5 times or less. And at first, when we realize this implication of this, that's an astounding fact. Most species on the planet actually have been observed a very few times according to our official databases. That's an astounding amount of rarity. And if we look at the functional relationship of these distributions, those distributions actually have a slope that is steeper than -1. In fact, it's -1.4, indicating that proportionally there are more rare species than we would expect, say, by neutrality and some of our other ecological niche models. Okay, so what we wanted to do is further explore then these patterns. And it turns out that there's a huge literature on these distributions in the microbial literature. And it's also known as the rare biosphere. Apologies for the typo up there due to PowerPoint transitions. And what this literature has told us is that if we actually look at another form of a way to plot this relationship, so this is a relationship between rank abundance, this is within a community or in big meta-communities, and the total number of observations per species, a large fraction of the species hang out in these rare tails. That is, most species by far are rare. And it turns out, thanks then to this literature on microbial diversity, if you actually sequence these rare species, what you actually find is that the majority of novel genes and the majority of novel functions reside in the tails and not in the most common species. And so many of these rare taxa, it turns out, tend to have these important ecological roles in microbial communities, these rare genes and these rare functions. if I can just pull out kind of one kind of quote then from one of these papers, this rare biosphere, which is the term, is very ancient and may represent a nearly inexhaustible source of genomic innovation. Rare taxa may have had a profound impact on shaping planetary processes. So it turns out when you sequence microbial communities and you look at these really rare taxa, and you look at the genetic and the gene composition and the functional composition, it turns out that in those tails, that's where the majority of functions, novel functions, reside. So, a deeper question. Why do we have all of these rare things on the planet, right? Well, we can return to our plant data and ask, where are rare species? Where do they tend to occur? Well, it turns out that rarity occurs in a very limited number of areas on a global scale. So when we ask where these rare species tend to accumulate, they tend to accumulate into what we call rarity hotspots. And it turns out that these rarity hotspots are really special places on the planet. The best predictor of rarity hotspots is the past climate. And it turns out that these are areas of the planet that have been the most climatically stable over the last 21,000 years. These are climatically stable regions of the planet. And this is consistent with the idea that these are areas then that have basically provided kind of protection and bastions then of diversity because the climate then is stable, presumably sheltering them then from extinction. Now, we can begin to explore a little bit more about rarity hotspots, and the implications. From the rare biosphere idea, our expectation was that there should be some sort of overlap, including at the global scale in terms of biosphere functioning. So, do rarity hotspots overlap with hotspots of biosphere functioning? The tentative answer, at least from our analyses, appears to be yes. So recently we worked with Martin Young's group, and we wanted then to overlap areas of global biodiversity with also areas of importance for global carbon and global water. So we took then information, our best information about variability in carbon stocks of terrestrial landscapes, as well as fresh water sources. And we wanted then to overlap all of this information to at least be able to say, in areas of biodiversity or rarity hotspots, combining then with global maps of carbon density, water, fresh water availability, and so on, do we actually find a correspondence? And in general, we do. So the global areas of conservation importance, if we want to prioritize high areas of biomass, which corresponds to these biodiversity rarity hotspots, as well as global important regions associated with high carbon stocks, okay, so not only carbon sequestration, but also in terms of productivity, as well as fresh water sources, it turns out that hotspots of this biodiversity, these rarity hotspots, also contain between 60 to 85% of the estimated total global carbon stock, and 66 to almost 90% of all the clean water then on Earth. Again, at the biosphere level scale, and overlap then between rarity, biodiversity hotspots, as well as global carbon and water. So, with that basic information in hand, we can then ask, well, what is the future going to be like? And what can we possibly expect? And what are the conservation implications? So we went ahead then and looked at regions then that currently have these biodiversity hotspots having to do with rarity. And then we looked at this ratio of future to historical velocity and temperature change. So in general, if regions then are blue, that means if we compare the past climate change that has happened out of the last 25,000 years ago to the future temperature change associated with RCP 8.5, which is the worst case climate change scenario, it actually turns out that compared to the past, the high the temperate and high Arctic regions are actually going to not experience as fast of change of temperature and climate than these tropical regions. So relatively, tropical regions then, compared to the past, are going to be experiencing a much faster rate of change. And if we then overlay on our biodiversity hotspot map this color scale of, in terms of the rate of change of climate from current day to the future, the redder the scale, the more red you go, the more climatically outside then these rarity hotspots will be from their current climate. And in general, what we find is that for the most part, a lot of the tropical rarity hotspots are going to be experiencing this accelerated rate of change well outside of the current climate that they're currently experiencing, which would be worrying because these are where rare species are, which are most prone then to potential extinction. Okay, so to conclude, as a biodiversity scientist, it's an exciting time because we now have access to then data and new ways, methods and new theories coming online by which we can begin to test and assess and then view biodiversity at this scale. So, rarity is common, exceptionally common. In fact, it's so common that it's the norm, okay? Most species are rare. And this rarity appears to be due to non-neutral processes. And rarity tends to occur in these really unique areas of the planet. But we also know that climate change and also human impact is going to be currently and in the future increasingly disproportionately impacting these rare species. And so I want to kind of conclude with the need for a theoretical approach in terms of understanding really what these rare species are doing and what they mean. And this very nice paper by Cyrille and colleagues proposed that we should think about rarity, this rare biosphere, not so much in terms of ecosystem functioning right now, but in terms of resiliency in the future, right? We know that the common species are the ones that dominate ecosystem flow and everything now. But the rarity, the things in this rare biosphere, that's the resiliency of the planet, right? At both local, but presumably also at global scales. And so these different curves then represent the increasing importance of this functional reservoir, this rare biosphere. And depending on the amount of uniqueness then in that tail of rarity, will influence then, as biodiversity then begins to erode in the future, how quickly things could potentially collapse. And for me, I think that is the potentially biggest insight. It's the resilience, right? It's the rare things then that are going to be influencing them, the resilience. But we don't know what those curves look like, which one will be on. And I think that's what this conference is in many ways about, right? Is biodiversity erosion going to result in a collapse, sudden collapse, or resiliency to a point of you can no longer hold out? And if we're on the top line, D, that could give us some time. Okay, so with that, I think I'm over, but thank you to an enormous number of folks and institutions and collaborators. So thank you.
Thank you for the very stimulating talk. Maybe I can play the devil's advocate. What botanists like to do, and I have one here, is to create species. So
I'm sorry, as to what?
What botanists like to do is to create new species. So every time there is a petal that is a bit different than a new species. So how sensitive are your analysis to the number of species, or to this, in another way to put it, if these species, which you consider as rare, are not actual species, but just, variants, and that you can cross them with another species which is not that far away.
Sure.
How does this affect the analysis you did, the first one, for example? I suspect that you will have a curve that is not bending at the bottom, but just straight. And it will be difficult to make the difference between the three
main hypothesis that you put forward at the beginning.
Yeah, so the question is, but these rare species, are they really new species, different species? Are there some sort of, error associated with that? In fact, that's the exact question that really worried us at the beginning. Are these real things, real species? Are they maybe taxonomic oddities? Are they maybe not really distinctive, not really true species, and so on? And so actually, I would encourage you to read the paper because we deal with that issue in quite some detail. And we parse the data in many different ways in order to address that. So the quick answer to your question is that it doesn't matter. It's not important. What was really fun is that one analysis that we did is that we randomly selected about 300 of those really rare species. And then we took those names out to the taxonomic experts of the world, the Missouri Botanical Garden, New York Botanical Garden. We found the experts on each of those different groups, mainly families and a few of the different genera. And then we asked them to, a bunch of different questions about those rare species. Are they, is it really, a real species? Is it an old name? Is it, maybe is it actually really common and that maybe it's not really, collected very well? And the overall majority of all of those names is that, yep, those are real species. Yep, they're rare. Indeed, your classification, is correct.
You mean morphological species?
Sure.
Yeah. The point is that you can cross many species of plants.
Yeah, I don't understand. Yeah, sure. I mean, hybridization is a real thing, so that doesn't preclude anything that was shown here. Yeah.
Thank you, Brian, for the fascinating talk. Hey, I sometimes have the feeling that we as biologists have a bit static view on how ecosystems function. So what you say is we have to conserve the current rarity because they provide unique functions. I say that we should conserve the future rarity of the new species entering the system because that provides your resilience. What do you think about it?
I mean, in general, I don't disagree with that statement.
See, in your last map, you show that the environment is going to change tremendously. You said it's going to impact the rare species. But the point is that these species maybe will not be hanging on because they are maladapted to the future environment. So you should have new rare species coming into your system and become dominant. So the question is, should we be worried about holding on what we have that is rare? Or should we stimulate new arrivals to be rare in the community and then become dominant?
Yeah, and so, can we predict the winners and the losers in that rare tail, right? So if that rare tail is the rare biosphere, that is our potential reserve, right? In terms of, these then are taxa that could be potentially rare in the future, be very important, for various different ecosystem services, and maybe even have that, that cancer drug that we, right? And so that's the excitement about the science of rarity, right? So can we, go out and look at this rare biosphere to say something about what then are the genes, the traits then that are going to do better into the future, right? And so, between us, we can come up with a list of traits and functions and life histories that are going to do better in a highly variable, more drought-prone, hotter world, right? In general, we know the set of traits and functions and life histories that tend to do best, right, in that future. And so that also means then that there's a set of traits and life histories and functions that are not going to do very well. Okay, and if we are interested in saving all the parts, by that means, then species then are going to be needed. But we also know that the traits and the functions and life histories then that add to enhanced ecosystem services, right? And so this is the science of rarity. I think we're still, we're there at the forefront. We're trying to figure that out.
Yeah. So you've been showing us some global distribution of rarity, and I was wondering if you've been playing with the scale at which you actually do that analysis and how the slope of the distribution vary with the scale of your sampling.
That's a great idea. You should propose that in a working group, and then we can work on that. Yeah. You know, no, that's a great idea. Yeah. Yep. Yeah. I was really intrigued with Anna's talk about this conflict between the global scale analyses and local scale, and that we have to work out those connections between the two. So I mean, you're absolutely right. I mean, I was like nodding my head with everything, and so we're at the point now where we can begin to do that.
Yes, thanks, Brian. I have actually two questions. The first one you almost answered, and the second one was: have you looked at the functional space of these rare species as compared to the other ones?
Yeah. That's a great question, Eric. You should propose that in the working group. Yeah. So, all right. So here's the problem with rarity. We don't really know much about these rare things, right? And so we don't really have, a lot of traits. There's a lot of, we talk about the data matrix and the NA matrix, the, missing data. Yeah, to be a trait ecologist and work on rarity is literally a nightmare, because you have tons of trait data out there, but nothing for, the rare things. And so, it would be wonderful to be able then to look at where these numerically rare things kind of fall. And this is in the working group, this is one of the tensions, is that, when we want to talk about functional rarity, we run into this ecological and evolutionary law of commonness and rarity, right? By far, the majority of our biodiversity is rare, that we don't know anything about. And so when we go out and sample the world, just by chance, you tend to sample the really common ones. And if you do hit a rare one, you often don't have, a lot of other additional, information, associated with that. And so what's been really fascinating for me in terms of thinking about the science of rarity is how we, really, how can we, take all our various different data sets and everything that have these big issues and holes of information that we're missing, and try to best fill that in. But it would be really good to look at, just functionally, if these rare things are doing anything important. I would encourage you to look at this rare biosphere microbial literature, because what I'm very envious about is that we can now sequence a lot of these rare things. And what they're finding is that long tail really does house the majority of gene diversity out there in terms, and also differing functions as well.
Hi, Brian.
Mm-hmm.
Thank you. I have one question. Actually, I was curious about the fact that in the places where you find these rare species, that the climate has been stable in the last, I don't know how many times we looked at it. And then, especially in these places, we see that future climate changes are impacting more. And that for me is a little bit contrasting, actually.
Yes, and it is contrasting. And that's what's, for me, particularly scary about those results, because it does indicate that you think about climate change as also relative then to the species and to the kind of historical climates then those clades and kind of individuals that have experienced. And so if our analyses are correct, it indicates that these rarity hotspots are going to be disproportionately experiencing rates of climate change that they haven't experienced, at least over the last 20,000 years.
So that would mean also that the previous climate change that happened in the world would affect differently these hotspots, or different the places in the world than this climate change.
Correct.
Okay.
That's the implication. Yeah.
Thanks.
Thank you.
Okay, thank you.
Thank you. I'm very, very happy to be here. I know everybody's thinking about cocktails, so I'm going to try to make this as interesting as I can. So I'm going to be talking about the functional consequences of marine megafaunal extinctions. Oops. So who is a marine megafauna? In very general terms, they are the biggest animals in the oceans, and that includes mostly sharks, my favorite, marine mammals, and seabirds, but also other species, other clades as well. And marine megafauna has always been around in this world, even since the geological past, which is what I study the most, the geological record. And I spent a lot of time about trying to understand one particular species, the megalodon. I'm going to talk about that in a minute. Until very recently, actually, maybe 2016, there was not a formal definition of marine megafauna. So everybody was using it in a very general way, sharks, marine birds, and cetaceans. But recently, there was another more formal definition of all the animals in the oceans that are over 45 kilograms. One of the most interesting or easy things to think about marine megafauna, at least from a paleobiological perspective, is that a lot of the species that we have today established in the shallow geological time, around 2 million years ago. Shallow for me because I think about 20 million years, but 2 million years ago, a lot of the species that we have today first established, or the assemblages. Traditionally, though, it was thought that a lot of these big animals were kind of very resilient to environmental changes. So for example, here, I'm showing this graph from a paper from Harnick in 2012, where they show that even though there was a very violent sea level oscillations in the last 10 million years ago, a lot of marine megafauna clades survived and nothing happened to them. One of the species that I'm most interested in is one of these extinct marine megafauna, the megalodon. You can see how big a tooth can be. This animal, it can be 20 meters of total length. So it was a massive, massive animal that lived in the oceans. It's very long extinct. So don't believe what you hear in the Discovery Channel that they say that it may be alive today. But the reason why I'm so interested in this species is because it was an apex predator, a cosmopolitan apex predator. And from ecology, we know that the consequences of losing apex predators are catastrophic. We know that there can be cascading effects affecting many levels in the food web. However, this is based on local extirpations and declines, because at least in the marine realm, we still have all of our apex predators. We still have our orcas, we still have our gray white sharks. So we don't really know what happens once we lose these big predators forever and globally. And that's what I thought, that the megalodon was a great system to understand the ecological consequences of extinctions. So I spent my entire PhD a bit obsessed with extinction of these species. Why, when, how it became extinct. So I studied these questions from many different angles. And because I only have 20 minutes, I'm going to try to be very brief. But basically, we tried to test one hypothesis, or a few hypotheses. One was that it became extinct because the water became too cold. And the species was a warmer water animal. And therefore, when the oceans became too cold, it went extinct. So we did, we reconstructed the entire fossil record of the species from its origination to its extinction. And also, we tested how the distribution changed through time in relation with climate. So we used climatic niche models. Long story short, we found one, that megalodon was widely distributed around the world. Here we have presence, this was a video, but it's a PDF. So presence in blue and absences in orange. And we did this model through time. And we saw that it was widely spread distributed throughout its entire geological time, or evolutionary history. And once we did the species distribution model, we saw that actually climate, or at least sea temperature, didn't really affect the distribution of these species. It could live in cold waters and in warm waters. Very similar to the great white shark. The great white shark is widely distributed in the world. It doesn't live in the poles, sure, but other than that, it can live in cold and warm waters. And that makes sense because there's a lot of evidence suggesting that the species was mesothermic. So it was able to regulate the body temperature of the most important organs, the brain, the stomach, the eyes. So of course, living in cold waters wasn't like a big deal that was going to drive this extinction. In fact, what we found that contrary to these climatic drivers, the decline of the species was more correlated with the appearance of new competitors. For example, modern orcas or the modern gray white shark, and also with the decline of potential prey, marine mammals. But of course, the question that I wanted to understand is, or the question that I wanted to answer was, what were the ecological consequences of this extinction? And I couldn't understand this by only focusing on one species. So of course I needed to understand first, if Megalodon became extinct alone, or if it was part of a global extinction event. And the reason why we thought this was a worthwhile question to answer is because we knew from previous studies that there had been a decline in the diversity of cetaceans around the same time. And also some charismatic species of marine megafauna were known to have gone extinct around the same time. Believe it or not, this is a giant aquatic sloth that lived in the waters of Peru. And we also know that became extinct around the same time. So to answer this question, we assembled a database from the entire Cenozoic, so the last 66 million years ago, after the extinction of the dinosaurs, to the last 2 million years of the marine megafauna. We downloaded it from the paleobiology database, which is an open source, public, free online resource where you can download fossil occurrences. It's worth noting here that the voluntary worker scientists maintain this data set. So for example, I spent a lot of time during my PhD entering data to this data set. And also we use Pirate, a revolutionary software that is that runs in Python, therefore Pi for Python, and Rate for rates of extinction and origination that was developed by my colleague Daniele Silvestro. And I say that it has revolutionized the field because it accounts for many biases of the fossil record. It models preservation rates and sampling rates. So it accounts for these biases. This is what we found. During the last 66 million years ago, we have an extinction peak in the Pliocene. So this is the time where the Megalodon became extinct, and also the Pleistocene, but mostly the Pliocene. But this is the entire marine megafauna showing at this extinction peak in the Pliocene. When we run the same analysis in a more continuous time frame, we see that's actually the late Pliocene where there's a peak of extinction. So indeed, there was an extinction event amongst these species. Oh, this was done to the genetic level. Different clades suffer different extinction intensity. Mammals suffer most losses. Around 50% of the mammalian genera became extinct around this time. And for example, sharks only 10%. But overall, the marine megafauna lost 36% of their genera from the Pliocene to the Pleistocene. In this graph, I'm showing in dark blue coastal species or coastal genera, sorry, and in light blue oceanic. And the reason why there's more extinctions in the coastal environments is simply because the marine megafauna live in coastal environments. That's where we have most of the productivity and where they're going to find most of the food. So of course, we're going to have more extinctions there. But of course, again, the question is, what are the ecological consequences of this extinction? At the time, I actually came here to Montpellier for the first time to try to see how can apply the functional diversity approach to the fossil record, because at the time, there hadn't been any studies that had done this like that yet, using a functional space. So we did this. We gathered the marine megafauna assemblage from the Pliocene and the Pleistocene, that is before and after the extinction. We assigned functional traits, body size, diet, thermoregulation, etc. We built the functional space, and we measure functional richness, the difference in the extent of the volume of the space before and after the extinction, a disturbance. And we also assess functional redundancy in terms of number of species per functional entity, or the number of species that have unique trait combinations. After we corrected for sampling size differences and run 100 simulations, we found that after the extinction event, we lost 15% of the functional richness, of the functional space. There was also a shift, a lack of overlap between these two spaces, the Pliocene and the Pleistocene, of 20%. We also lost 7 functional entities or unique trait combinations. So let's back up for a second. 36% of taxonomic extinction, 15% of functional extinction, 7 functional entities lost. So of course, this was not by no means a mass extinction event. But when we compare it with other studies that had used a similar approach, we found that those numbers were very striking. In this paper by some colleagues from the Naturkunde Museum in Berlin, where I did my second postdoc, they found that after the mass extinction of mollusks, where they lost 90% of the diversity, only 1 functional entity was lost. So how come the marine megafauna is losing so many? Well, they're functionally unique. They have very low redundancy. In these histograms, I'm showing the number of species per functional entity, both before and after the extinction. And as you can see, most of these entities are filled by 1 or 2 species. And if you think about it, it makes sense. There cannot be many species of blue whales or many species of megalodon. The species are very unique. There's only 1 with those traits. So I was thinking about this difference between big animals and small animals, giant sharks and tiny mollusks. What's the difference between them? And what makes some perhaps more resilient than others in terms of ecological changes or functional changes? So I talked about this idea with my colleague Christine Bacon from Gothenburg. And she said, you know what, there's an extinction event in the Caribbean where we lost 70% of the benthic communities. Let's try that idea with those communities and see what we get. So we got together with Alex Siska and Austin Hendy, who had collected a massive database of fossil occurrences of Caribbean mollusks. This extinction took place around the same time as the marine megafauna, but it affected mollusks and only in the Caribbean. A lot of people thought that this had something to do with the rise of the Isthmus of Panama. Some others think that it's because of the glacier interglacial periods. Regarding of the cause of the extinction, the fact is that there was something going on at the time that affected benthic communities regionally very deeply. So we use the same approach, functional diversity richness and functional entities, to see how functional redundancy changed. We collaborated once again with Daniele, who created this pirate software, and indeed we found extinction rates were very high in the late Pliocene, just like with the marine megafauna. This has been proposed earlier, so this wasn't new. We quantified the number of species that had gone extinct, and we found that 50% of the molluscan species became extinct. But in terms of functional richness, as you can see here, there were barely any changes. Functional diversity remained the same, even though 50% of the species went extinct. So Daniele went a step further and developed what we call, I think, the multi-trait extinction dependent model, which assessed the role of traits and trait interactions with extinction rates. So all the functional traits that we had assigned to the different models, we correlated them with extinction rates. So you have diet, like habitat, and so on. And we also included redundancy. Even though it's not a trait, we were very interested in how redundancy correlated with these extinction rates. And actually, we found that that was the key cause or the key determinant of this extinction. Redundant species, redundancy was the one factor that would explain better the extinction rates. And within redundancy, species that were highly redundant had more chances of going extinct. So yes, of course, we're not going to see a lot of changes here because a lot of species that were going extinct had other that didn't, that had similar traits. So far what we can say that from these 2 extinction events, it's evident that the marine megafauna is highly vulnerable because they have low redundancy. They're very unique. Even though they had a moderate taxonomic loss, they lost way more functional diversity compared with Caribbean mollusks that had a way more intense taxonomic loss, but the functional diversity losses were almost nothing. And of course, I mean, the redundancy was very different. The mollusk had at least 8 species per functional entity, whereas marine megafauna had 1 or 2. So obviously, the next question that comes after this is like, if we know that the marine megafauna became extinct around this time and they were so unique and so on, what caused this extinction? So the usual suspect was the differences in the very violent sea level oscillations of the time. As you can see, this is it had been proposed by multiple authors that have modeled sea level changes through time and have found that in the last 10 million years, but especially the last 5 million years, sea level changed dramatically, violently. So I collaborated with Sara Varela, and what she did is that for each sea level change scenario here, I think the resolution is 100,000 million years, she calculated how much area was left after those sea level oscillations, and only the continental shelf area, so over 200 meters of depth. So remember that the continental shelf is not constant. Some parts of the world have a wider continental shelf than others. So we actually quantify how much area was left given this. And this is what we found. This is area, not sea level. In the late Pliocene, around the time of the marine megafaunal extinctions, we lost a lot of area. So these marine megafauna saw themselves in an environment where less area, coastal habitats were available. And not only that, but it became very, very variant, 3 times more variant than before, and also 30% less overall. And there were times in that the area available were 50% less than before. So we proposed that habitat loss was a key cause of this extinction event. And this was corroborated by some analysis that John Griffin did, in which we assessed the role of traits on extinction rates. And we found that not body size or diet explained these extinction probabilities. It was thermoregulation, with homeothermic animals, mesotherms and endotherms, having higher chances of going extinct. And the reason why this corroborates the area loss hypothesis is because homeothermic animals, because they regulate their body temperature, they have way higher metabolic demands than ectothermic animals, and therefore they need more area. When area is not available to find enough prey, they get themselves in trouble. So in geological time, when you have an extinction event, often there is a phase of recovery. So under normal circumstances, you would expect for this marine megafauna to be recovering today. But of course, that's not the case. As we saw in previous talks, we're entering, and it has been argued that we already entered a 6-mass extinction event. So this marine megafauna went through an extinction event recently, is now being hit by a massive extinction event. For example, pelagic sharks are suffering a drop of 70%, and overfishing and bycatch is deeply affecting other species of marine megafauna. So the question that then we ask is, what is going to happen to the future of the marine megafauna? So we established a database of the current modern marine megafauna that we live in our oceans today. We assigned different functional traits and built the functional space of this current marine megafauna. And instead of looking at the past and see extinction rates in the past, we just did forecasts in the future. So we use IUCN statuses, and we forecasted 2 extinction scenarios. One, what would happen to them in 100 years, given the extinction probabilities correlated or related to this extinction risk statuses. And another scenario in which we assume that all species that are currently threatened will go extinct. This is what before we did these scenarios, Camille and Fabienne, who are all very own Montpellier people, help us with this functional diversity analysis. We created the spaces. The key message here is that we have some species that are very weird. Some of you guys were asking me about who are the distinctive species of the marine megafauna. Here we have the whale shark, the Atlantic fur seal, the right whale, and so on. But most interesting to me, at least, is the fact that even though fish are the most diverse ones, it's mammals, the ones that occupy most of the space. Now let's go back to the extinction scenarios. So remember, 100 years and all go extinct. If regarding the extinction scenario in 100 years, we predicted that around 20% of the marine megafauna can go extinct. But if all threatened species go extinct, we could lose around 45%. Now let's focus on this scenario. Once we calculated functional richness, given this extinction scenario, we found that we could be losing 50% of the functional richness of the space. And this is significantly different than what you would expect by chance. So we're definitely targeting certain distinctive species. I'm going to skip the uniqueness for now, but it's going to be bad. We also did the same for each clade. And the bottom line here is that sharks are the ones expected to suffer the biggest losses, both in terms of taxonomic richness, but in terms of functional richness. We could be losing 80% of the functional space of sharks if all of the species currently threatened go extinct. So we proposed a conservation metric called FUSE, functionally unique, specialized, and endangered. Of course, very much inspired by the EDGE metric. So why functionally unique? Because we don't want to, we want to avoid leaving gaps in the functional space. For example, we don't want to give, we don't necessarily want to leave a big gap within the functional space, which will be left if functionally unique species go extinct. And specialized, because specialized species are the ones that are at the edge of the functional space. And if they go extinct, then there's more functional space being lost. So we combine these two functional metrics with IUCN status, and we propose what are the most fused species that are both functionally unique, specialized, and endangered. The green sea turtle, the dugong, the Julian's golden kelp, I always forget the name of the species in English, sea otter, and the giant clam are the most functionally unique, specialized, and endangered species. And because I'm running out of time, I'm going to skip the work of Ariana, my master's student, who mapped these metrics through time. Just to say very quickly, I don't know if Anna's still in the audience, but some of the areas that we are highlighting here as having a lot of functional richness or a lot of top few species are not necessarily the ones where the species are restricted to. Like the whale shark, for example, will be living in all of this area. It's a widely distributed species, but there are certain areas that bring together many distinct species, even though they have a worldwide distribution. And because I think I'm very much out of time, I just want to quickly thank my collaborators and my funders and you guys for listening. Thank you. Great presentation, thank you. I was wondering if the difference between the mollusks and the megafauna could be how you measure the traits in the fossil record. Because I'm assuming that you get a lot of information from the skeleton of a shark, even like teeth and things like that. But from a snail, I mean, can you get really meaningful traits about diet and things that could you be just underestimating how much functional diversity you lost for the mollusks?
Yes, a great question. The way that the accessibility of the data for sharks and mollusks wouldn't have much of a difference in the fossil record. In case of sharks, we only have teeth, really, not real skeletons. And the good thing about the mollusks is the fact that they are benthic communities and they have shells, so they tend to fossilize more. So in that sense, I guess we have kind of the same level of resolution, but we chose different traits because, like, mobility, for example, was an important one for mollusks. Some are sessile and some are not. But for sharks, most of them are mobile, so we wouldn't use that. So we made that decision based most on what they do in ecosystems as opposed to how much information we can find. But yeah, the resolution in terms of that would be very similar.
So thanks for your talk. So going back to Anna's talk, so here you really don't have really arguments about ecosystem functions. It's more, I would say, response threats. So use threats to.
Yeah, it's more.
Yeah.
Sorry.
Okay.
Yeah, it's more about what they do. Is the trait that we use? We're more thinking about what they do in ecosystems, really, and we try to link actually for the marine megafauna and the Anthropocene. We try to link every trait to their functions in ecosystems. So for example, mobility will be related, or migration, for example, for the Anthropocene one, we didn't use that for fossils, will be related with nutrient transport and so on. So
in your case, it really brings more than evolutionary distinctiveness.
Say that again?
Focusing on traits instead of just phylogeny or evolutionary distinctiveness brings more in terms of
results and interpretation. So why is it interesting in your case?
Yeah, well, in the case of the marine megafauna, because it's a multi-clade assemblage, so we have the marine mammals and the sharks, and the cutoff is based on size. We didn't use the phylogenies because we're only using certain parts of the tree. But for the new projects, and stay tuned, we're now using a clade approach. So we're comparing different facets of biodiversity, including phylogenetic diversity and functional diversity. But for the marine megafauna, that was particularly challenging because we're not using the entire tree of life of any of these clades.
I wonder how much big size is a big matter, it's a big confounding factor, because if you're big, you have not that much chance to speciate because you cannot have that many big mammals in the ocean. And if you're very big, you're very distinct. Also, you have this distinct diet. And also, if you're very big, you have a particular demography, so maybe you're more extinction prone. So maybe it's all confounding, so can you rate the parts?
Yeah, that's actually the whole point of my new 5-year program. We're trying to bring together those questions into the role of extinction rates and phylogeny into how they end up working out with the traits. So in a few years, I may have an answer to that. Yeah.
So thank you for your talk. If I understood correctly, in the case of sharks, it was that they became extinct because they had redundant traits, right? Redundant functional traits. And I was wondering with the presentation of Brian, for example, but also with the results at the end of your presentation, you're focusing again on the conservation of species which are more rare, maybe. So I was wondering, what's the conclusion?
Yeah, no, it was the mollusks that became extinct, that the extinction rates were correlated with redundancy. The most redundant ones were the ones that had higher chances of going extinct. And in terms of the marine megafauna, most of the functional entities in general, it's very low redundancy, and we didn't test the role of redundancy in extinction rates. But going back to the question of rare species, so what we're doing with this conservation metric is combining these functional indices with IUCN statuses. So in a way, that's a way to think about rarity, if you will, based on the fact that a lot of these IUCN statuses, the criteria are population decline and also geographic range, but whether or not it's abundance, rarity, or looking at it in terms of a restricted geographic range is more about what is the status of it right now. So we're combining, we're not thinking about it as in rarity, but most of like extinction risk based on whatever criteria you're using to assess that, and combine it with the functional indices. So what are the species that are already identified as a priority because they are going extinct, plus the ones that are contributing disproportionately to maintaining the functional space. I don't know if that makes sense.
Yeah, okay.
Cool.
Thank you.
Thank you.
Thank you.
So for the last talk of the day, so really happy to introduce David Mouillot. Most of you know him very well. So he's a professor at the University of Montpellier and interested by a lot of questions related to biogeography.
So yes, I work at the Marbeck laboratory, mainly in marine biology. So I will talk today about the biogeography of functional rarity. But before that, let's go back to the 40 years ago. Most of you were not born when Radnovich introduced the 7 forms of rarity. So you can see in this very nice cube, you can have different aspects of rarity depending on 3 dimensions. The first one is the local population size. So some species can have small or large populations. Some other can be very widespread or very restricted in range size. and some other can be very specific or are very wide habitat. And in this cube, you can have 8 scenarios, and there is only one where the species is very common. This is the gray one. Otherwise, each species has at least one aspect of rarity. You can see this cube in this plot as well. You can see the 8 scenarios, so it's a bit boring, but I would like to focus on the green one, which are the common ones, so large population sizes, broad habitats, and this one, which is the opposite, where you have populations size which are very small in specific habitat and so on. And the main hypothesis of Rabinovich is that species being in this last class should go extinct more rapidly, or should be more vulnerable to many impacts. Must say that I struggle a lot to find evidence about that since the Rabinovich paper. I found this one, which is great, because we look at more than 6,000 marine invertebrate genera over 500 million years. And for all their taxa, what they found is very clear. I mean, for 3 groups, so gastropod, beavers, brachiopods, when you look at the odds ratio, you can see for abundance that the large population sizes or the small ones, there is no significant difference. So it means that either you can be very abundant locally or not abundant at all. There is no effect at all on extinction risk. When you look at range, it's very clear. You have a very significant signal for the 3 groups. So all the species belonging to these groups are more vulnerable to extinction where they are very low range size. And for habitat specificity, the same thing. We have not a very clear signal. So it means that for all these taxa, range size is the main aspect driving extinction risk over time. I found very few other evidence or studies like this. So it means that since we are biased, since we tend to publish only the most exciting results or the most pessimistic, I think that very few guys found very convincing results about this kind of patterns. I just got this one, which is very recent, perhaps less exciting, on fireflies, where they show that beyond rarity, what they found is that some aspects like diet specialization or poor dispersal ability were more related to extinction risk than the aspect of rarity proposed by Rabinovich. I prefer this one in current biology some 5 or 6 years ago, where they show that finally what drives species to extinction are traits, and some which are very valuable for people, like, for instance, horns or other aspects, and also animals with very big sizes or extreme traits. So somewhere, traits matter a lot for extinction, drive extinction. Some are very old or extreme, and we must think that most of the species play very unique roles in ecosystems, like top predators. So traits matters. So it's why we did these papers in 2017. So it's a kind of turning point towards functional rarity, where we try to combine 2 aspects to define an ecologically rare species. So first, so many functional species, but I like a lot this one with the traits here you can see, and the distribution of many terrestrial vertebrates. The first aspect for sure is that you get very rare traits or very unique or distinct traits, like these big guys here or the very small ones as well here. What we call functional distinctiveness. So very easy. You have been immersed on that since the early afternoon. The other aspect we combine is geographic restrictiveness. So once again, very easy. You get species which are very widespread. So when they combine low distinctiveness, like this antelope, with a widespread distribution, you are very likely, when you go outside in savanna, to see these traits. So very similar traits from other species or this species, which you can see almost everywhere. So in this case, this species is functionally common, very easy. At the opposite, you have this one, which is only in one place, maybe a cave, I don't know. And these species are very distinct in the functional space. So when the species combines both aspects, you can tell without any doubt that it's a functionally rare. So this is the basis of what we did in many further studies based on this concept. So you can classify species. I will focus only on two groups based on quartiles. So here you have geographic franchise and functional distinctiveness. And the ones which are functionally rare are those ones, the red ones, with distinct traits and low franchises. At the opposite, you have the common ones with very common traits and a very large franchise. So you can do the same thing for birds and mammals, terrestrial ones. So today I will have four main objectives to know which are the ecologically rare species of vertebrates, where they are on land and sea. We are very lucky because we got 2 recent studies on that, dealing with that. To know whether they are very threatened or protected by some conservation strategy or measure. And an exciting topic, it's on islands, because we have many extinctions, and to know whether the new species, alien species, get the same traits, can make a kind of turnover, or have a very limited set of traits compared to native ones. So for vertebrates, let's go back to these great papers from some guys in the room here. So you have seen that before. So we can see that 2 functional species for terrestrial birds and mammals. And the goal was just to extract the ecologically rare species among all these guys based on franchise and trait distinctiveness. So it's a very arbitrary cutoff, but we got some unread mammals and birds, which we define as ecologically rare. We did some over-analysis showing that if we change this threshold, we got similarly the same results. And the first attempt were to locate the hotspots to know where they are mainly based. So you got this map that everybody knows to show the species richness of mammals and birds on Earth. And you can see the hotspot that we all know, like tropical South America or Eastern Africa or Southeast Asia. But when you focus on the richness of ecologically rare species, you get totally different patterns. I mean, it's very widespread. You have many locations, even in very northern countries, which are hotspots of ecologically rare species. And you can see that most of ecologically rare species, for me, it was the most surprising result when Nicolas came to me the first time with that result, is that finally very few pixels, very few areas on Earth have all these rare species. And maximum value can be up to 12 or 28, which is a lot, like you can see for Madagascar for mammals, or like the Andes for birds. So all these ecologically rare species tend to pack some very restricted areas. They were particularly present on islands. They are very hotspot of ecological rarity, most of these islands. So then we had a look to check whether they are protected, whether they are under threats. The very obvious and very trivial result is about the IUCN category. So you can see here the percentage of species being threatened or least concerned on the IUCN red list. And you can see for the mammals, the ecologically rare species and the common ones. So there is no surprise since we include geographic franchise into the game to see that most of rare species tend to be threatened compared to common ones. It's not a big surprise. What is less trivial is that we can see that for target achievement in terms of coverage by protected areas, you can see that the ecologically rare species are underrepresented. Most of the time, they do not have at least 10% coverage by protected areas, compared to common ones, which are not so well protected, but a bit better protected than the ecologically rare species. This is not trivial at all because there is no link between coverage by PAs and the fact to be ecologically rare or very common. exactly the same for birds when you see that the ecologically rare species tend to be underrepresented into the global system of PAs compared to the most common ones. So we can say that they tend to be less protected on average than ecologically common species. I like this recent study as well, where these guys had a look at 9 islands where we had experienced many extinction in the recent past due to humans, which arrived, came with new species and makes some local ones disappearing. So what they check, they look at this even greater functional space for birds, and they look at the relationship between the quantity of invasive species and the extinction, and you can see that there is a very clear link with, on many islands, a proportional arrival of new species, alien species, compared to local extinction. So we have many local extinction, like Hawaii, and many new species coming. And the question is whether the new species, the one which replaced the native ones, came with the same traits or with more diverse traits or very common traits. And what they found is very striking, because the red one is the functional diversity, like morphological space in 3 dimensions, for the birds before the arrival of humans. So you can see that all the red box plots are quite high on the 9 islands, from New Caledonia to Hawaii. So it's a very common pattern. The yellow one is after the humans came, so it's before, without considering invasive. So you can see that the drop is huge. So with extinction, we talk about perhaps less than 5% of species going extinct for birds. You get a huge drop of functional diversity just after this marginal taxonomic extinction. So many species disappeared with very extreme traits. And what is very interesting is that you remember that on most islands, when species disappear, many new came in, alien species. So the blue box plot is after the arrival of invasive. You can see that despite this increase, sometimes very high in specific diversity, species diversity, you do not increase a lot the functional space or functional diversity. You are still very far from the red ones. It's very impressive, and sometimes even lower or at the same level as the yellow one. So what they show very nicely is that this is before extinction. So the native, when humans came, tend to be on the edge of the functional space, the one which got extinct. And when the alien came, they just came inside the functional space without coming with new or extreme traits. So there is no replacement. There is just poorer or lower, more restricted functional space after invasion. So there is no, I would say, resilience of functional diversity. So we have this in the sea, we have this on land. Even after a drastic change, taxonomic richness is not changing a lot. You have simply species extinct, new ones coming in, and there is no functional resilience. We got that on coral reefs. We got that in many places. Even after establishing marine protected areas, there is no functional resilience. You never go back to the pristine states. So we got back in the Pacific, for instance, because we know what is the pristine state. So now we go back to marine fish. This study came out some weeks ago. So it's great to see these results. So an even more beautiful functional space for fish, where you can see distribution of fish according to nice traits like schooling behavior, position water column, diet. And they use exactly the same strategy as Nicolas MOUQUET for birds and mammals, terrestrial ones. They got the occurrences in fish bays, they got the traits, they estimated functional distinctiveness. At the end, they just split species into many categories. This one is the most interesting ones. They are the ecologically rare species. So I mean, distinct traits and very narrow range size. And then they extracted the richness of these species being in this category for teleost fish and for sharks. And like us, I will be rapid on that. Like us, they found that they are very clustered in space. So here you can see for bony fish on coastal ecosystems only, where you see the density of species being classified as ecologically rare. And you can see the same thing when you control for species richness. So it's even more impressive. Like you can see that these hotspots are not located everywhere, but also in some very temperate or even Antarctic areas that concentrate most of ecologically rarity on coastal ecosystems. This is quite similar for sharks or elasmobranchs that occur almost everywhere. But when you look at the ecologically rare species, you can see the spots which are very, very narrow, very located, like South Africa, like Southern Australia. So we don't have these strange forms, so big sizes or very small one or strange diets, always in the tropic on the cradle of speciation. But we got these guys close to temperate areas or close to polar areas. It's we got something consistent with this paper of Rabowski, which was very controversial some years ago. Guys found faster speciation rates outside the tropics. It was a very surprising result, and we found something quite similar with very strange forms and functions. It was a source of novelty, these very temperate or polar areas, at least for marine fish and elasmobranchs. So very interesting result to see that we have many hotspots very far from tropical areas for these strange forms of fish and elasmobranchs. And when you look at the coverage by marine protected areas, you can see that we are not so good. We have many protected areas across the sea. We are close to 8% coverage, the target being 30%. But you can see that on average, we only cover half of ecological rarity across the ocean. The same number for teleost fish and elasmobranchs. So it means that with the ongoing decline of sharks, Katarina showed that very nicely that sharks display the highest functional distinctiveness across the oceans. We get a substantial loss of functional diversity if we do not protect very rapidly all these critical areas where sharks tend to thrive. But they are the last sanctuaries, perhaps, where you can see these ecologically rare attributes, traits, or species. I cannot finish this presentation without talking about this new nice paper from Nicolas. He will talk about about that if she wants. The goal was to rank species, reef fish species, according to their beauty. The public was in charge to give marks to these beautiful fish by pairwise comparisons. So they did that. It was a huge work. They did that on the web. And then using machine learning algorithm, we classify all the species according to their beauty or being ugly or very beautiful to the eyes of people. So we are a bit far from ecological rarity. But the goal to me was to try to link this beauty, this link to people, this emotional link to this fish with functional distinctiveness. So first, when Nicolas did that, I was sure that the most beautiful would be the most distinct. So I was very confident that at least this fish, this distinct fish, could attract media coverage, conservation interest, or simply a big human interest for their case. But I think we were surprised, Nicolas, me, and the others, to show that when we plot on the functional space the aesthetic value between the blue ones, ugly ones, to the most beautiful ones, you can see the beautiful on the left, very clear, and the one we don't like so much to see. No surprise. And you can see this kind of triangular relationship when you have on the x-axis functional distinctiveness, on the y1 the aesthetic value. You can show that the most distinct are not so nice, or even the ugliest one. So it's a very depressing pattern, saying that they are very distinct, they are threatened, not covered by protected areas, and nobody cares. So, so there is no link, no emotional link between these distinct species and the human brain. There is no particular link to influence decisions or conservation or even scientific interest towards these fish, which are certainly key players in ecosystems. So it's a nice end to this depressing talk, perhaps. So my conclusions. So first, we can see that functional rarity emerge almost anywhere across the tree of life, across the oceans, across Earth. So we are still weak in explaining such patterns, why they emerge in almost all families, in islands, in the Antarctic, in Hawaii, in many places. So there's a lot of work to do on that. We know that this functional rarity is very packed, impressively packed into some small areas of region, which is good for conservation. It's very intriguing as well to see the packing, special packing of this functional rarity. The other message is that this functional rarity is still poorly covered by protected areas and is not attracting to humans. But we have a big ambition to cover 30% of Earth, including land and sea, before 2030, which is almost tomorrow. So we hope to include this kind of aspect, functional rarity, into IUCN guides or with NGOs or all these organizations making conservation strategy by now. And the last point perhaps is that when you lose some local species with distinct traits, there is no, to my knowledge, example where you come back to a very functionally rich ecosystem. So both in the sea and on terrestrial ecosystems. So when they are vanished, it's hard to get back with the species having the same diversity of traits. Thank you.
I was curious about the link you found between functional rarity in the maps that Hrabowski have found for respiration rates in fish, and what you think is the connection, if there is any, classic diversification mode that may be linking respiration rates with rarity, and if you know of any empirical evidence of respiration rates being triggered by rarity.
I'm very happy to be here. No, it's a very critical question, I think. I think the better guys, the best guys in the world on that question do not agree at all. Because we are supposed to have seen a very explosive radiation of forms and functions on coral reef, mainly coral triangle. So it was a very strong surprise to see other spots of differentiation for extreme traits. So it's a kind of a totally opposite result that they got. So I have no idea whether these two mechanisms can occur at the same time, or if Hrabowski paper can be under threat very soon, because I know some responses will come out very soon on this paper as well.
Yes, for the islands, I guess most islands have no native mammals. I wonder if that to me is a big problem, because the community are completely changed, the ecosystem is different, because living with cats and rats for island birds is different. So I guess we cannot recover the diversity.
Yeah.
It's not a rule mistaken, but
yeah. It's more surprising in the open sea, because by connectivity we should recover after the local extinction or local extirpation of some species, but we did not. So it's more surprising in the sea where all is connected. So there is no barrier to colonization, and most of the time, after local extinction or local extirpation of some species, it's hard to recover a functional space.
Thank you very much, because I'm not at all in the field of functional ecology. I'm more a conservation ecologist. And I'm actually working as a co-chair of a group of specialists for the IUCN. So we are setting goals for what we want to do in this group. And I see, well, we should go into functional writing. Yeah, why not? But what I think a lot, and I haven't seen, and maybe it's just because I haven't read fully your papers or understood the field, is about data quality. And it is kind of the nerve of war when you're working on conservation, having the good data to go into this mapping and understanding the patterns, spatially or temporal. And of course, you can use models, but I don't know how much you guys use these prediction models of spatial distribution or whatever, as Catalina was saying. There are also lots of questions about bias on the data. And I don't know, because I found it so crazy that you have like lots of data for the global scale for most of species that you've shown. I work on insects, and most of the time you have scarce data. You might have opportunistic data from Atlas and GBAF, so you don't have like schemes from throughout years that are very standardized. And so this is the kind of data that I have to do this kind of assessment later for IUCN and stuff like that. And also about traits, because I suppose that you guys, one of the limits is having the traits, and not only the traits, but linking these traits to functions, and these functions may be to ecosystem services. So my question, broad question, or at least like discussion, is about how much these methods and approaches and measures handle this bias about the data and the scarcity of data, and how much do you know that you have all the traits that you need to go into these spaces and stuff like that. Because I think that for conservation, it's something that maybe me or other colleagues will be afraid to go into these approaches, because we're missing already lots of information and data about biological records and taxonomy and stuff like that. And this is like a new challenge to integrate to, but that is super amazing. And I think that all that we've seen today is great, but we have to see how much can we really go into that. So it's very open question, discussion. Sorry.
I try to be brief. For fish, we are very bad about occurrences, to be very clear, because in this vast ocean, it's very hard to get very reliable data. So all that we show here, it's very interpolation a lot. So we are very poor in predicting where fish really occur. We may have many surprises. So it's why we need new methods and you need more empirical data. It's a disaster for occurrences. I am a bit more confident for traits, because we begin to infer them quite confidently across species, the tree of life. We begin to get intraspecific variability. So for me, we accumulate much more knowledge and it becomes even more predictable. So I am a bit more confident in traits than in species occurrences. But it's a big problem. I totally agree. And on the coast and in the deep sea, in the ocean, the knowledge of fish abundance and presence is not yet resolved and will not be in the next 10 years.
Thank you.
I think, in fact, this question is a perfect timing to open for broader discussions if you want. So maybe some of you want to answer to data questions, Brian or Aurel or Brian Mitner. Maybe we can open just for 10 minutes. I think it's a good questions that's open for general discussions about do we have the data to perform some conservation exercise? Yeah.
Sorry, I cut it off a little early.
Sorry.
Regarding the data availability, I can actually answer that question. I've been looking at that recently, and at least for plants anyway, the areas where we're missing geographic data, or where we're missing genetic data, or where we're missing trait data, all line up very well, like very strong correlation. Like, so it's unfortunate because our gaps are all in the same place, at least for plants. I don't know if anyone has a good feel for where that is for other taxa. But for plants, the gaps are all in the same spots.
I have a practical question about referring to the point about missing data on the rare species. Let's say I want to contribute to this and measure some functional traits and other traits. What would be based, I guess it's a question to the speakers and maybe others, what would be, based on your analysis, do you have suggestion about the best way to select species? Because of course we can't study more. What priorities and what criteria would you use for species selection to contribute to this rarity framework?
So I mean the priorities of the species to measure in priority in terms of traits, you said.
And criteria for species selection.
It depends on the questions, but for instance, when looking at the ESN status, so we have so much non-evaluated species. For sure, based on evolutionary distinctiveness, it's a good candidate to measure traits first, because we can expect that they are also functionally distinct. So this is a way to target those species. Other answers maybe from others?
What about geographical locations?
Sure, this is the way, but we found that we have no relationship between geographic scarcity and functional distinctiveness, so it won't inform about that. For most of the species, we have some information about body size, for sure, it's a key trait. So based on that, you can focus on the gradient of body size. It would be a good way to target species along this gradient, so it could be an example.
Thank you.
Other questions, feedbacks? Not sure we convinced you to go into this functional conservation paradigm, but maybe it's worth doing. Other questions or feedbacks? More generally, yeah.
I thought that for modern species, I was going to get a lot of data because they're modern, they live here. And it turns out it was maybe as bad as or worse than for fossils. So personally, my reflection is that even though we are in the age of information, there's a lot of databases, there's a lot of models and tools to infer traits, to model geographic ranges. To me, the reflection that I take from it is that we still need to go back to that primary data, you know, taxonomists, people going to the field and measuring things. I think that for a lot of animals, especially marine, we don't really have a lot of basic information. What's the size of this fish in this part of the world, or where does it occur? So I think that even though we live in this age of information, there's a lot of value still on primary data, and students, especially from the global south or poor nations, going measuring insects and invertebrates and fish, and taking this data that was lacking, that even if we have the most amazing tools and models and sophisticated software, there's nothing like actually, you know, taking this data. So I have been reflecting personally a bit a lot, and I've been making use of a lot of techniques, but nothing like going back to the field, boots in the ground, and collecting information. And I think that perhaps we can make more of an effort to go back to that kind of way of thinking. I mean, me included. I haven't been in the field in a long time. It would be great to contribute further to these problems of not having enough data. So yeah, just that.
So I would like to thank you all, and I will let Hélène conclude with a few words. Thanks a lot.
So the conclusion of the afternoon would be: read the appendix and the supplementary materials. It's very important. So I just want to make a 4 round of applause for 4 groups of people. I think we had a wonderful afternoon. So I would like to thank a lot again our wonderful speakers. Well, Cyrille, Anna, Brian, Catalina, David, thank you so much for your talks, and it was amazing. So second group of people, I would like to thank again the FRB team, and especially 3 people who haven't been thanked so far. So Robin, which is in the room somewhere, Pauline and Claire, who are here. They have worked with the FRB, with Maud and Marie-Claire for the organization and the communication. And before, during, and after the meeting, they will be working. So thank you for them. And speaking about communication, I would like to thank as well the University of Montpellier team, because they have recorded this afternoon, and you will be allowed, thanks to them, to attend again this meeting on YouTube. So thank you. And finally, I would like to thank all the FRB and CESAB postdoc, because they feed some of you, they hold the mics, they run on the stairs sometimes. Thank you, Davey. I don't know where you are. And they guide you, and I think without them, the day would have been less interesting. So thank you very much. Thank you very much. So Aurore, Davy, Bruno, Aaron, which is somewhere as well, Babacar, and Ali.